Okay, so we're going to get started this afternoon. It's going to ramp up into some fairly advanced terrain here. We have the first talk of the afternoon from Bliss Chapman from the University of Illinois, who's filling in for his boss, Sanmi Koyejo, who unfortunately couldn't make it. But I'm looking forward to seeing Bliss's take on synthesizing fMRI, applications, promises, and pitfalls. So take it away, Bliss.
Thank you. All right, hello everyone, and welcome back from lunch. My name is Bliss Chapman, and as he mentioned, I'm here from the University of Illinois at Urbana-Champaign, and I'm super excited to talk to all of you today about generative adversarial networks and some of their applications in cognitive neuroscience. So to give some context to this talk compared to what we've seen so far today, Sage talked about neural networks for brain age prediction. Hannah talked about neural networks for segmentation. Grace talked about neural networks as a way to model cognitive processes. And now I'm going to be talking about using neural networks for synthesis of new data using this framework called generative adversarial networks. All right, so I hope you all had your cappuccino at lunch, because we're going to start with a game, and I need your participation. So one of these rows of faces are fake people. They're generated by a deep learning model. And the other row has faces of real people. And so I want all of you to raise your hand if you think the top row are the fake people. Okay, probably about 20, 30% of you. And now I want you to raise your hand if you think the bottom row are fake people. Okay, there's about 10% of you who don't know what you're doing here yet. That's okay. And it turns out that the top row are fake people and the bottom row are real people. Thank you. And now we're going to play the same game. So one of these images is a synthetic X-ray, and one of these images is a real X-ray contrast from a radiologist practice in Chicago. And so I want you to raise your hand, again, if you think the image on the left is the real X-ray. Okay, 40%-ish. And now I want you to raise your hand if you think the image on the right is the real X-ray. Okay, cool. Now before I give it away, can someone who picked the image on the left tell me why they chose that? Just yell it out. Is there any particular feature of the image that you noticed or picked up on? Okay, interesting. And now can someone who picked the image on the right tell me why, or tell me anything they picked up on? Okay. All right, so the image on the left is fake, and the image on the right is real. And if you didn't do too well in these games, you shouldn't worry, because you aren't alone. When experienced radiologists were asked to choose which of these X-rays was real and which was fake, images produced by a GAN, by this deep learning model, were identified as real 39% of the time. And notably, there was quite a spread in how successful these radiologists were in distinguishing these real samples from these fake samples. There was one radiologist who preferred GAN images over 80% of the time, and there was another who could identify real images successfully almost every time. And so today, we're going to be talking about synthesizing fMRI data. And so as a final game, let's see how well all of you do in guessing which of these two sets of brain volumes are real. And these are brain contrast statistic images, which are thresholded using whatever the Nyland defaults are. So raise your hand if you think the brain volumes on the right are real. Brain volumes on the right. Okay, not many of you. And now raise your hand if you think the brain volumes on the left are the real ones. Okay, seems like most of you are sick of this game. All right, congrats to all of you who got that right. In the rest of today's session, I'm going to introduce GANs, which are the deep learning models that generated those synthetic samples. I'm going to discuss how to use GANs to synthesize fMRI data specifically. I'll showcase a few applications of this technology in cognitive neuroscience, briefly mention some challenges, and then finally wrap things up with a tutorial showing all of these concepts in action. So without further ado, let's jump into generative modeling. So mathematically, we can think about a data set of examples, x1 to xn, as samples from some true data distribution, p of x. And in this figure, the blue region shows the part of an image space that with a high probability contains real images. And these black dots indicate our data points. So each of these black dots is an image in our data set. And if that was confusing to you, you can think of an image space as sort of all possible images. So if you were to enumerate pixel by pixel and try every possible value, that would generate all possible images. However, only some of those images would make sense. A lot of them would just be, random noise of some sort. And that part of the image space that would make sense is shown by the blue area in the figure. And of course, then these black dots, which are representing our actual data points, are within that blue figure. And what we want is a model that describes a generated distribution, p hat of x, that is similar to our true data distribution. However, reasoning about these kinds of problems in high dimensions is hard, so I'm going to first review what a generative model looks like in low dimensions to build some intuition. So let's suppose we had a bunch of two-dimensional data scattered around in a grid, and we were tasked with estimating what is the underlying distribution of this data. And we could start by selecting some parametric family. This data looks to me like it's bivariate normally distributed, so that's a reasonable first choice. And then we could simply find the parameters of that distribution that maximizes the likelihood of my data. But why do we care about this? Why is it useful to find the generating distribution in the first place? Well, it turns out that if we can recover the underlying distribution of our data, we're the king of the world. So think back to your intro to statistics class for a moment, and let's suppose you had some distribution of data. Maybe this distribution represents the number of car sales you make in a month, or maybe this distribution represents fMRI scans of healthy subjects. And then your friend comes along, and they bring you a new data point, a new fMRI scan. They might want to know how different from usual is this fMRI scan? Or in what ways is this fMRI scan different from a typical fMRI scan? Or how are two populations of fMRI scans different? Or how many samples would I need to reliably detect a difference between two populations of fMRI scans, etc.? If we know, or if we can estimate well, the generating distribution of our data, these are the types of questions that statisticians have built tools to answer. So on this slide, I show an example of hypothesis testing when our generating distribution is a univariate Gaussian. So given some hypothesis about a new data point, or some populations of data, we can evaluate the likelihood of that hypothesis being true using our understanding of that underlying generating distribution. And it's important to recognize that all of these same intuitions hold in high dimensions as well. However, the standard parametric families and their fitting techniques don't really scale well to complex high-dimensional data like brains. So modern techniques use neural network-based approaches like variational autoencoders and generative adversarial networks, which I'll go ahead and introduce now. So if we think back a few slides to our initial mental picture of what a generative model should do, we want a model that describes a generated distribution that is similar to our true data distribution. And a generative adversarial network, again, tries to describe this generated distribution by finding a function that maps from a simple unit Gaussian to the generated distribution. So to sample from my generated distribution, I would simply take a Gaussian random variable and pump it through my neural network, and that's easy. However, notice that I am not defining an explicit probability density function for my data. And this makes computing the likelihood of a new data point very difficult. And so for this reason, GANs are part of a class of models called implicit generative models. They are implicit generative models because they don't explicitly define a density function. Now, how do we go about estimating theta, the parameters of this function that map me from this unit Gaussian to the generated distribution? Well, if I think about this intuitively, what I want are parameters that make my generated distribution similar to my true data distribution. And given a unit Gaussian sample, my learned function, my generative model, can transform it into a sample from the generated distribution. And if I was a domain expert, maybe I look at fMRI images all day, I might be able to look at that generated sample and tell you whether it looks realistic or not. In other words, a generated sample is bad if it is easy to say it does not come from the true data distribution. And similarly, if the sample is good, it is hard to tell whether or not it came from the true data distribution. I'm going to repeat that one more time. So if a sample is good, it is hard to say whether or not it came from the true data distribution. And so the GAN framework captures that intuition by using a classifier to learn a loss function. And this classifier neural network is often called the discriminator, and it is trained to determine if generated samples look real or fake. And so now our two neural networks play a little game. The generator neural network is trained to fool the discriminator, and the discriminator neural network is trained to avoid getting fooled. So if the generator network is successful, then the discriminator network will perform no better than random at determining whether a generated sample is real or fake. So zooming out to see the entire GAN architecture at once, first we draw a sample of random noise from a Gaussian distribution. And then we feed that Gaussian random variable through our generator network to produce a sample from the generated distribution. Then we feed that generated sample to the discriminator, and it makes a prediction, real or fake. Then a loss function is computed for the generator based on how well it did in fooling the discriminator. And similarly, a loss function is computed for the discriminator based on how well it did in determining whether the sample was real or fake. And then both networks are trained with gradient descent based on the gradients of their respective loss functions. Uh-oh. Okay, there we go. And the craziest part of this idea is that it actually works. So here on the right, you can see some pretty pictures generated from GANs. And I want you to remember that none of these bedrooms actually exist. These are all figments of some neural network's imagination. And the success of this technique has made it wildly popular within the ML and AI community, and tons of progress is continuing to be made in this area. Just to get a sense of the hype, here's a graphic showing the cumulative number of GAN papers by month and a sampling of GAN architectures being explored. And this curve just keeps going up. So now that we have a sense of how GANs work, let's talk about GANs for fMRI. In this session, we're going to use the Brainpedia dataset available publicly on NeuroVault. The dataset has around 6,500 statistical parametric maps covering 45 total tasks and 19 cognitive labels. And here's the architecture of the GAN model we'll be using to synthesize fMRI data. So in the orange box on the left is the generator network, and in the blue box on the right is the discriminator network. And they're essentially mirror images of one another. A couple minor points differ from what we've talked about here so far. The first is that because we are synthesizing the entire brain volume rather than just a 2D slice, this model uses 3D convolutions to produce 3D images or volumes. The second minor difference is that our brain data actually has labels indicating what cognitive task was being performed. And so we feed these labels into the generator network along with the random noise. And then the discriminator network enforces that the sample generated by the GAN, or by the generative model, is a realistic sample given those cognitive labels. Okay, so not just a realistic sample overall, but a realistic sample of those labels. And then that signal encourages the generator model to learn to synthesize conditionally based on what label was provided. And the result is the ability to synthesize realistic brain data with user-specified labels. And you can even generate combinations of labels that weren't present in the original dataset. But how do we know if our synthesized data is any good? Well, my first instinct would be to hire a bunch of domain experts to look at it, maybe some of you out there. And that's a pretty common approach, quite reasonable actually, although it gets expensive quickly. And this is also fairly qualitative evidence of how good your data is, and why should I trust the neural network in your head rather, or any more than I can trust, the artificial neural network of the discriminator. I want hard evidence. So if you want quantitative evidence that your synthetic data is awesome, then you have to be a bit more creative. The NeuroSynth image decoder is an open source tool that allows you to quantitatively compare a brain volume to images in the NeuroSynth database. And here, we use it as a tool to evaluate the quality of our synthesized data. So given a brain image synthesized for specified labels, we would like to see high correlation with the analysis types of those labels. Another approach to quantitatively evaluate our synthetic samples is to use a subset of our data to train a generative model. Then, use synthesized data from our generative model to train a classifier. And then evaluate that classifier's performance on a test set of real data. If our synthesized training samples are high quality, meaning they come from the same distribution as the real data, then we should expect a classifier trained on those synthetic samples to perform similarly to a classifier that was trained on entirely real data. And in fact, for our model, we see this happening. As we increase the number of synthetic images the classifier sees during training, the performance approaches the accuracy of a classifier trained on entirely real data. And the cool thing about this evaluation technique is it can actually be reformulated as an application of our model as well. So instead of training a classifier only on real data, we can instead train a classifier on all of the available real data, plus as many synthetic samples as we care to generate. And as you can see in this table, this yields performance improvements in our classifier. So some of you may be thinking right now, how is it possible that adding fake data improves classifier performance? And this is a hot research topic right now, but here's one hypothesis to give you some intuition. You can think of the synthetic data as filling in the data distribution between the real data points. And when you do that, it acts as a regularizer of sorts, which improves our classifier's ability to generalize. And notably, traditional generative models, like a Gaussian mixture model, do not have nearly the same success as neural network-based approaches do for these sort of downstream classification tasks. In this table, you can see how augmenting the training set of a classifier with synthetic data from our GAN model consistently outperforms data augmentation from more traditional techniques. Data augmentation is just one use case for this synthetic data. As I mentioned earlier, we can generate combinations of labels that aren't present in the original data set, and some of my latest experiments are actually exploring the idea that maybe we can use synthetic data to determine sample size requirements for new experiments where we haven't even collected data yet. Now, before I move on to the tutorial, I want to briefly mention that these techniques have their fair share of problems. I won't be talking about them in detail here, but I think it's intellectually dishonest to not at least have a slide about them. So if you're interested in diving into the GAN literature, you should be sure to Google these terms or come talk to me after the session. All right, so to wrap up the slide portion of this session, synthesizing fMRI data is exciting, and it's a promising technology for accelerating brain data analysis. The early results are promising, but there is a lot of work left to do in this area. For example, using generative modeling as a tool to explore individual differences in neuroanatomy, extending our techniques to model time series of brain volumes rather than just a 3D snapshot, improving the evaluation techniques, working at higher resolutions, etc. All right, so with that, I'm going to try to jump over to the code here. If you want to follow along, you can follow along by going to the website and clicking on the tutorial, or you can just follow this link here. Okay. All right, so over in the tutorial here, I'm going to start running. The first thing I'm doing here is downloading all the Python packages I need. And then I'm going to unzip them. And then download the data set. This will take not that much time at all because I already did it on this machine. It should take about 30 seconds if you're doing it out there in the crowd. I'm going to unzip it. Okay. Then I'm going to fetch the list of requirements, the Python packages I need, and then install them. and go ahead and import all the packages I need here. So I am using PyTorch, which is actually a different framework than most of the tutorials you've seen so far today. So far, I think you've seen Keras and TensorFlow. PyTorch is another deep learning framework. Fix some random seeds so that the results are the same every time. And scroll, scroll, scroll past the download. All right, so I'm setting up some paths here for where the data will be stored. This data is actually going to be downsampled. So instead of using the full brain volume, I'll be downsampling it to 1/4 of the original size. And I'll be using a batch size of 128. This is a fairly arbitrary selection. It was just made based on how big my GPU memory is. Then I'm going to load my data set into memory. And I'm going to go through the exercise of plotting an example data point. So if you aren't familiar with data science or data science techniques, this is a pretty good practice. Anytime you get a new data set, you should, of course, explore it before you try to apply any of these advanced methods to it. Here, I'm going to do the most basic thing you can do, which is just try to plot out the first data point in the data set. So my brain data has this shape. This is the number of samples in my brain data. And this is the X, Y, and Z. And this dimension here is just extra to make it easier to interface with the PyTorch API. Let me scroll down here. And so here is what an example looks like. And notice that each example comes with a bunch of tags. So in this case, this brain volume is tagged with the cognitive process label calculation, visual words, and visual. And this will be important because remember, we are synthesizing conditionally based on these labels. So when we go to actually train this model, we're going to want to see that we are synthesizing examples that are not just realistic brain images, but realistic brain images of that type. Okay, now getting on to training the model. I'm going to be focusing today on what's been different than what you've seen so far, and mostly what's different than what you've seen so far are the way that the GAN model is trained, because we have two networks that we're actually training simultaneously. All right, so here are the two networks. We have the generator network and the critic network. This is another word for the discriminator network. And again, they're basically mirror images of one another. And again, because I'm focusing on the training process here, I'm abstracting away the exact architecture of these behind these two constructors here. And this model takes a long time to train. So I've actually trained this model for around 200 epochs overnight. And I'm actually going to be loading it in now from a checkpoint. So if you want to do this at home, just know that you should probably have access to a GPU if you want to actually train this model in full. Okay, so I'm going to train it for an extra 5 epochs here to show you how this works. And if you are into math, this is the loss function that the GAN model is actually trying to optimize. There's a couple terms here. Basically, these terms are describing what I told you in the slides. One of them is penalizing the discriminator based on how well it did in determining whether a sample is real or fake. And the other term is basically rewarding the generator based on how well it did in fooling the discriminator. There's one extra term here, which actually comes from the distance function we're using to measure between probability distributions. This is a much more subtle topic. If you're interested, I'd be happy to talk about it in the Q&A or afterwards. Okay, I'm defining some helper functions here to generate noise. And this is the most important function of this session. This is how a generative adversarial network is actually trained. So there's a couple steps. The first thing we're going to do is enumerate over every batch of data in our data set. And then we're going to train our critic, our discriminator network, to distinguish between real and synthetic brain samples. Okay? So to do that, we are first going to generate a synthetic data batch for the specified labels. And we're going to train the critic, right in this line here, given a real data batch and a synthetic data batch, plus the labels for those corresponding batches. All right? That will give us a loss, and based on that loss, we will then train the critic network. The generator network is, again, trained to fool the critic, and so what we do is we gather a sample of noise, feed it into our generator network to get a synthetic data batch, then feed that synthetic data batch into our critic network along with the labels. Then based on the critic predictions, we compute a loss function. That's what you see in this line here. And based on that loss function, we then train the generator network. All right, and then there's a bunch of code here to actually visualize the training procedure. And I'll go ahead and run the training now. So this is just a sample of the full data, so it takes maybe 30 seconds for each epoch. And I'm going to show you one epoch here and then move on to how to synthesize whatever label combination we want. And as you can see right here, this model has already been trained for 241 epochs. All right, boom, done with the first epoch. There's two separate loss terms, remember, one for the discriminator and one for the generator. And it's going to try to visualize here examples from both the true data, given some labels, and the synthetic data, given those labels. All right, so once again, let's go over here. Here's true, visual, right hand are the cognitive process labels. And the synthetic data for those two cognitive process labels looks like this. All right, and there's a whole bunch of samples of these you're more than welcome to play around with. But what I want to do here in my last couple minutes is actually do a quick live coding exercise showing how to sample the cognitive process labels that you might want. So I'm going to jump over to here and grab some code snippets. So the first thing we're going to do to generate a new sample from this network is to grab a sample of random noise. So this could be from a uniform distribution or from a Gaussian distribution. In this case, I'm doing Gaussian. I'll just show you what that looks like. Oops, I gotta stop this up here. Okay, so here's our sample of noise. And the second thing we're gonna do is decide on what label we want to synthesize. So here, in this case, I've decided to synthesize an example of the visual cognitive process. And to do that, I need to get this label into a format that the network can actually understand. And so what I've done here is do what's called a multi-hot encoding. A one-hot encoding, which you may be familiar with, just means that for every type of label, I'm going to represent that as a vector of zeros with a 1 turned on in a certain position. Okay, so as you can see right here, if I turn on the 1 in the 3rd position, that corresponds with the visual cognitive process tag. And all that's handled here in this method I wrote. All right, so now we have our label and we have our noise. The next step is to synthesize a sample from the generated distribution given that noise vector and the encoded label. Go ahead and do that now. And then finally, I want to see what that looks like. So I'm going to visualize that synthesized sample. All right, here we go. So here is a synthetic volume of a brain for the visual cognitive process label. And I might want to go back later and change this to be something like right hand. And if I do that, I'm going to run these all again. So generate a new noise sample, encode that label, and in this case, it's no longer the 3rd or 4th, it's now the 5th. 0 is turned into a 1. I'm going to generate a synthetic sample of that label, and then I'll visualize it once again. All right. Cool. Thank you all very much. If you have any questions, I'd be happy to answer.
Sorry if I may have missed this, but fMRI data typically evolves over time. Mm-hmm. How does the GAN take care of the time evolution?
Yeah, great question. So in this work, what I've been showing is simply synthesizing fMRI volume at a specific moment in time, so a snapshot. All right? And what I'd like to do in the future moving forward is to extend this to modeling how cognitive processes evolve over time. But that requires different types of networks, like Vince talked about earlier today, like recurrent neural networks and that type of thing. Does that answer your question?
Sort of. So are there recurrent GANs? Does that
yes.
exist?
Good question. So pretty much any type of neural network architecture you can think about, there is a GAN variant of it. So yes, indeed, there's an RNN version of a GAN.
And the models that you were showing, those were individual fMRI volumes, just one acquisition.
Right, yes.
Thank you.
Thank you for your nice, wonderful talk. I'm wondering if it's possible to generate all the true distribution of the data with GAN, with just limited number of fMRI sample data. Is it possible to generate all the true distribution of data?
Well, if the model works well, that's indeed what we're hoping to have happen. So what's happening right now is we're given a true data distribution, and what we want our GAN to do is learn to synthesize samples that we cannot tell whether or not it comes from the true data distribution. So what I think you may be getting at is there's many different noise vectors that you could generate and feed into your network. And actually, instead of sampling randomly from that noise distribution, you could actually sample in very careful ways to produce the kinds of output you care about. So if you really wanted to, you could do something like sampling a wide variety across some Gaussian distribution to see and try to understand what is that latent code really helping you understand about the model. Is that kind of what you're getting at?
Yeah, I see. Thank
Okay.
you.
If I understand correctly, the map that you show is like a Z statistic map. So I'm thinking, is it possible also like to generate like fMRI time volume? It may not correspond to any task or any kind of like resting state network.
Sorry, can you say that one more time? An fMRI what?
Like a volume, like fMRI time point.
Okay, and you want it to be not conditioned based on any respective like cognitive task?
Yes, and also basically similar to the first question that he has basically like you just generate resting state or task fMRI data itself.
Okay, I can maybe try to shed some light on this. We might want to follow up on this afterwards if I don't quite answer your question. So if I were to not feed in these labels that specify the cognitive process, what I would expect to have happen is the generator network should learn to produce samples from all of these different types of cognitive process labels. And back to the previous question, it actually might be the case that different parts of that simple unit Gaussian distribution might end up representing different types of cognitive processes. And they might be actually quite, clustered almost in that Gaussian distribution. What was the second part of your question again?
The second part of my question. I think I can follow with you later.
Okay, sounds good.
Thank you.
Thank you.
Hi, I was just wondering if you could use the same kind of approach to generate only good quality fMRI data, distinguishing it not from noise, but from some sort of imaging artifact or things like that.
Oh, yeah, good question. So, yeah, very good question. There are a couple papers in that area, actually. One interesting thing about all of these sort of deep neural network architectures for generative modeling is that some of them tend to smooth the output and some of them don't. So GAN is actually well known for producing sharp, realistic images, but there's another type of deep neural network architecture for generative modeling called a VAE, or a variational autoencoder. And it's been shown that variational autoencoders can actually smooth the output of a data set. So for example, if you have a bunch of images that have some sort of weird imaging artifact and you know what the imaging artifact is in each particular picture, you could train something like a GAN or like a variational autoencoder and penalize it based on whether or not the latent representation, the latent code, the Z variable, the unit Gaussian sample, actually has information about that artifact or not. Okay, so you could think of basically adding on an additional classifier that would penalize our model based on whether or not there's information about that artifact in some layer of the network. And then what you can do is you can say, I want to generate an image with that artifact or an image without that artifact. And so then what you might do is you might feed in an image with an artifact, turn on the switch that says produce an image without an artifact, and see what comes out the other side. And there's been some work in this area. You might want to look into adversarial variational autoencoders or something like a fader network. Those are both good places to start for that kind of approach. All right, thank you very much.
Actually, I got a question.
Oh, okay.
So thank you for the nice tutorial. I got two questions. One might be somehow overlap from the two questions, but I want to make it clear that, to my understanding, the generated images are all like contrast map. It's not raw ball signals. So earlier two questions are some of Z-score maps getting after SPM processing. So I don't think the generated one is the real like FMR ball signal. That's one question. And the other one is about the GAN training. So one of the challenges in GAN training is model collapse. So I'm just wondering, how did you handle that issue in training your model?
Yeah, great question. So the first one, I would have to check the details of the data set myself. I don't quite recall. For the second one, I actually have a slide. For those of you who don't know what he's talking about here with mode collapse, let's see if I can show this real fast. It's an important note. So as he mentioned, GANs are susceptible to mode collapse. And what that means is, if you think about what is the easiest way to produce a realistic brain image, what you could do is simply choose one brain image that you know is good. If I was the generator network and I was lazy, I didn't want to do my homework, I would just choose one brain image and then synthesize that brain image over and over and over and over. So rather than capturing the entire distribution of my data, I might just capture one data point and learn to synthesize that one data point really, really well. And there's a couple techniques that are employed to try to resolve this problem. In this model, in this tutorial, I didn't get into any of that, but I'd be happy to chat offline about techniques in that area. So in this figure, you can see the target distribution has a bunch of dots arranged in a circle. And as model training progresses, you can see that the GAN sort of zooms in on just modeling one of those data points rather than all of them. And so that's, the term for that is called mode collapse.
All right, unfortunately, we're out of time. Thanks for all your questions, and thank you, Bliss, for a great talk. So let's give him a hand. Next up, we have Anders Eklund, who has been parallelizing since before deep learning was cool and CUDA was the monster it is today. He's the first professor who's been on the stage for a little while, and at Linköping University, I hope I pronounced that right. You might know him from his work with broccoli or from the cluster failure paper, but he is also going to be talking about GANs. So, looking forward to the talk.
Okay, thank you for the introduction. So we have received some hardware from NVIDIA. So I will also talk about GANs, but not noise-to-image GANs, but something called image-to-image GANs. So I will start talking about what's the difference between these types of GANs. I will mention two different architectures for image-to-image translation. So the first one is called pix-to-pix, and the second one is called cycle GAN. I will then go through some different applications of image-to-image translation, like synthesizing T2 weighted images from T1 weighted images, synthesizing CT from MR, synthesizing PET from MR, and etc. I will also show one example of distortion correction and one example of abnormality detection. And I will end by talking a bit about different challenges. So one challenge is to go from a 2D GAN to a 3D GAN. A general problem in deep learning is that if you train a model on data from one scanner, it will not work as well on data from another scanner. And the last challenge, if you train your GAN on controls, can you then actually use it for deceased subjects? So the use of GANs has really exploded during the last years. So this is an overview from a nice review paper that in 2016 there was only a single paper, but then it really exploded in the last years. And the most common application is synthesis of new data, but you can also use image-to-image GANs for segmentation. You can use it for reconstruction, classification, and so on. It seems like the most popular modality is MRI, maybe because you have so many sub-modalities, but GANs are being used for many different types of medical images. So as Bliss mentioned, if you have a noise-to-image GAN, you always start by generating a noise vector, and then your GAN will hopefully produce a very realistic image. So this person doesn't exist, but it's just a very realistic synthetic face. And every time we send a new noise vector to this GAN, you will get a new realistic image. But image-to-image GANs are a bit different, that you don't send a noise vector, but you send an image to the network, and you get another image from the network. And this can be used for different kinds of translation tasks. So you can, for example, convert a horse to zebra. You can convert winter images to summer images. You can convert apples to oranges. So these are some examples from the cycle GAN architecture, which is one of the most popular architectures. So you can basically convert between any two domains that you can think of. And this can be before and after segmentation, or before and after distortion correction, or whatever you can think of, as long as you have two domains. And maybe you have seen that Snapchat has released a number of new filters that can actually do what they call a gender swap. So if you want to see a GAN in action, you can open Snapchat, you can apply this gender swap filter that will convert male to female or female to male. I'm not sure what the exact technique is that they use, but I'm pretty sure it's some kind of image-to-image GAN. And maybe you can tell me who these two persons were the original images, which person they represent. So who do you think this is? Yeah, so this is a female version of Donald Trump. And who do you think this is? Yeah, so this is a female version of Nigel Farage. So if you want to train an image-to-image GAN, you basically have two options, to use a supervised GAN or an unsupervised GAN. So you have images from two domains that we can call X and Y, and then we can train a GAN to convert from one domain to the other. So here it depends on what kind of data you have to start with. So if you have paired image pairs, paired images, you can train a supervised GAN that can learn from this joint distribution. But it's not always the case that you have paired images. So maybe you only have a number of images from one domain and a number of images from another domain, but they are not really related to each other. Then you can train an unsupervised GAN that also has to learn the joint distribution. So to show an example of this, this is one example of paired images where there is a real correspondence between the two domains. And here's one example of unpaired training images where you just, for example, have a number of photos and a number of paintings. And in some cases, it's not possible to have paired images. For example, for the Snapchat filter, you cannot have paired training data of men and women. It's not possible to have that kind of data. So we can think of it like this, that if you have a supervised GAN, you want to learn this blue joint distribution, and you get paired samples from this blue distribution. So then it's not very hard to learn the distribution, but for an unsupervised GAN, you only have samples from the two red marginal distributions. And from these samples, you have to learn the joint distribution. So an unsupervised GAN is a much harder problem, but you can use it for many more applications. So one of the first image-to-image GANs was the pix-to-pix. So you have images in the two domains. They need to be paired and registered. Then pix-to-pix can learn to translate from one domain to the other. So you can, for example, see that you can convert from a street scene to a segmentation of each pixel. You can convert a map to an aerial picture. You can convert day to night, edges to photo, black and white to color, and so on. So the main drawback here is that we need paired images and they need to be registered. So if we want to instead do unsupervised translation, for example, using CycleGAN, we need to learn this joint distribution from the marginal distributions, and this is a much harder problem. So we need some kind of regularization to handle this. So what is used in CycleGAN is that they add a cycle consistency loss. So if you take one image from domain X, you transform it to domain Y, and then back to domain X. It should be similar to the original image. And in the same way, if you translate an image from domain Y to domain X, back to domain Y, it should be similar to the original image. So this cycle consistency loss acts as a regularization to prevent that the training fails. So this is my only math slide, but just to define the loss functions. So we have, in CycleGAN, we have two generators and two discriminators. So we have one generator that goes from X to Y, we have one generator that goes from Y to X, and then we have the two discriminators. So the adversarial loss function we can write like this, that if the discriminator for Y gets an image from the Y domain, it should return 0 since it's a real image from the Y domain. If the discriminator receives an image that has been translated from the X domain, it should classify that this is a fake image, return 1, so that the sum here is 0. So this is the traditional loss function for training again. So then we need to add the cycle consistency loss function. So we start from some image from the X domain, we transform it to the Y domain, and then back to the X domain. And then we take the difference with the original X image. And the other way, we take an image from the Y domain, transform it to X, transform it back again to the Y domain, and take the difference with the original image. And then we use the L1 norm to get the scalar from these two different images. So the full loss function is one adversarial loss for each generator, and then this cycle consistency loss. And lambda controls how important is the consistency loss compared to the adversarial losses. As I mentioned, we're training two generators and two discriminators at the same time. So we're training 4 convolutional networks at the same time. And this is one example of what the discriminator and the generator can look like. So the discriminator is sort of simple with 4 convolutional layers. The generator is a bit more complicated. So since we have to start from an image, we first have some convolutional layers where we downsample the image. Then we have a number of residual blocks. And then to generate a new image, we need to upsample the result that we get in the middle, so to say. So just to show you one example, so this is the text from the CycleGAN paper that describes the generator. So they use a number of different types of convolutional layers. So for example, this C7S1 minus K is a layer with, where the filters are 7 by 7. DK is another type of layers where the filters are 3 by 3. Then they have RK for residual layers. They have UK for another type of layer. And then they can write out the full generator using this notation. And it might seem that would be very complex to write this as a code in Keras, but actually this code represents the full generator. So for each type of convolutional layer, a help function is defined, and then we can use that help function to add different types of layers. So this small for loop represents the 9 residual layers, for example. So you can write that very compactly with Keras. So these are just to show two of the help functions. So DK, it's a very tiny function that takes K as an argument. So how many filters you want in the convolutional layer, and then the filter size is set to 3, and then you apply some normalization and activation function. The residual layer is a bit more complicated, but the code is still pretty short. So if we look at the discriminator, it only has 4 convolutional layers plus the final classification layer. This is the corresponding text in the paper. So here they write that they use something called a patch GAN, which basically means that instead of directly classifying if the image is real or fake, they first classify every patch in the image. But otherwise, they only use 4 convolutional layers, which you can write like this. And then you can decide if you want to use the patch GAN or if you want to convert the final image to a vector and use a fully connected layer for the actual classification. But this is the whole code for a discriminator, so it's pretty easy to write in Keras. So let's look at some of the applications. So one of the first applications for MR images is to convert between different types of images. So you can maybe pretend that you have collected T1 and T2 weighted images for every subject, but maybe for some of the subjects you cannot use the images because the subject has moved too much. Then you can train an image-to-image GAN to synthesize T2 from T1 or the other way around. Or for example from T1 to T2 flare or from proton density to T2. So in this paper they compare a number of different approaches to perform the translation. And to produce a synthetic T1 or T2 weighted image can also be used to make the registration easier. For example, if you need to register a T1 weighted image to a T2 weighted image, that can be a bit difficult. But if you instead produce a synthetic T2 weighted image, you can register T2 to T2 instead. Or if you want to do segmentation of brain tumors and you only have T1 weighted images for some subjects, you can synthesize T2 and then combine T1 and T2 to perform the actual segmentation. So another application is to go between MR and CT. So in this paper, here they show how to go from an MR image into a synthetic CT image. And this is pretty close to the real CT image. So this can, for example, be useful in radiotherapy, where you need to plan how to treat brain tumor patients. So then you often need a CT volume to calculate the dose you need to treat the patient. And if you can avoid collecting the CT volume, that will save some time and radiation for the subject. So in this paper, they also show that they can go from CT to MR, which is maybe more impressive since the MR volume has more details inside the volume. So this is a good example of the cycle consistency loss. So you start with a real MR image, you go to synthetic CT, and then back to MR from the synthetic CT image. And this image shows the error between the real MR and the reconstructed MR image. So another application is MRI to PET. So in this paper, they used ADNI data, and they wanted to classify if a subject is healthy or has Alzheimer's disease. So for most subjects, there are MRI and PET data, but for some subjects, the PET data is missing. So they trained the GAN to translate between MRI and PET. These are the results for 2 subjects, comparing the real and the synthetic PET images. And then they show that if they add this additional data when training a classifier, that they can actually improve the performance compared to only using subjects that have real MRI and real PET. So here, the GAN, you can see it as a kind of data augmentation that you synthesize PET data for subjects who don't have PET data to have more training data for the classifier. This is one example from my research group where we trained again with T1 weighted images and fractional anisotropy maps from 1,000 subjects in the Human Connectome project. So we wanted to see, can we go directly from T1 to FA without collecting any diffusion data? And it seems like you can get pretty close to the true FA maps without collecting any diffusion data. So we sent these results to a conference, and then the reviewers said, like, okay, you have some nice results, but what is the real application here? So then we show that we can actually use this for distortion correction. So in the Human Connectome project data, you have diffusion data with 2 phase encoding directions, so left, right, and right, left. And they both have pretty strong distortions. So for example, with top-up in FSL, you can correct for this distortion. But the problem is that most data sets do not have two phase encoding directions, and it will double the scan time. So instead, we synthesized an FA map from the T1 weighted image, and this FA map has no distortions. So then we could do a nonlinear registration between one of these 2 original data sets to this synthetic FA map. And then we can produce a similar distortion correction without doubling the scan time. In another application, we we wanted to see if we can revert a face blurring. So normally you blur the face of every subject before you publish your data. So we wanted to know, is this face blurring enough for anonymization? So here the two domains are the original data without face blurring and the data after face blurring. And then we used CycleGAN to learn to translate between these two domains. And this is only a two-dimensional GAN, so it doesn't use any 3D information. It's only trained on sagittal slices, but still we can sort of recover the face. So since GANs are so powerful, maybe we need to redefine what we mean by anonymized data. So if we look into a bit more advanced architectures, so instead of learning many one-to-one mappings like T1 flare, T2 flare, and so on, why not use all the data when we try to predict a missing image? So this is the main idea of a color GAN, that uses a collaborative GAN to instead of converting from one image to another, you try to use all the available images to predict a missing image. So in this case, they combine the 3 types of images to predict image number 4. And then they show that this approach gives a smaller error compared to, for example, CycleGAN and another architecture called StarGAN. And another idea is to combine different GANs, since a noise-to-image GAN can be pretty difficult to train due to mode collapse and so on. So instead of directly trying to synthesize data, you can try to do it in 2 steps. So you can use a noise-to-image GAN to synthesize some object of interest, like a binary segmentation mask, and then you can use an image-to-image GAN to transform this binary mask into a realistic image. So this approach was used in this paper. So they first did segmentation of these retinal images to get binary segmentation masks. And then they trained a noise-to-image GAN to produce a new realistic binary masks. And then in the second step, they trained CycleGAN to translate between the binary segmentation masks and the retinal image. And these are some of the synthetic datasets that look rather realistic. And here the advantage is that you know that this is a perfect segmentation of the retinal image, since you have created the retinal image from the binary mask. And they also show that training with the synthetic data and evaluating on the real data gives rather similar performance compared to training only with the real data. So I will now cover a bit of the challenges that we currently have with GANs. So almost all the GAN papers you can read, they use 2-dimensional GANs both for synthesis and translation. But in your imaging, we have 3D and 4D data. So you can, of course, apply a 2D GAN slice by slice, but it will probably not give you a perfect 3D reconstruction. It's pretty easy to change the code from a 2D GAN to a 3D GAN, but the memory consumption will really increase a lot. So you more or less have 2 options to work on subvolumes or to buy a better computer. So the few papers that have used 3D GANs, and they work on subvolumes, and then they do the translation for one subvolume at a time. Training the GAN is what takes the most time, but once you have trained it, you can run it on the CPU for the actual translation. So if you think back of the code that we looked at, so we had the conv2D to perform the 2D convolutions. We have conv2D transpose that's needed for the back propagation and upsampling 2D. So to change the code to 3D, we only need to replace these functions with conv3D transpose, and upsampling 3D. So it doesn't seem very difficult to go to 3D. But if we think of what one convolutional layer looks like, it's pretty common to have something like 256 filters, and then you need to store all the filter responses somewhere. So for one image, that's like 64 megabytes, so it fits on the GPU. But if you have a volume, then it's suddenly 16 gigs. And there are very few consumer graphics cards where you can store 16 gigs in memory. And this is only for a single convolutional layer. So either you have to work on subvolumes or buy some more advanced hardware. For example, the NVIDIA DGX station, you can have 128 gigs of graphics memory, but that's pretty expensive hardware. So another challenge is that if you train again on data from scanner A and then you try to test it on data from scanner B, it will not perform as well. And this is not specific to GANs, but it's a problem in deep learning. So to show one example, we trained a GAN on 1,000 subjects in Human Connectome project, and then we tried to apply it on a completely different data set. And then it doesn't perform as well as before. So there is no easy solution to this problem, but what you can do is to, of course, train the GAN on data from many different scanners, or you can train another GAN to translate between scanner A and scanner B. So you first apply that GAN, and then you need to apply your original GAN. But then you need a lot of images from scanner A and scanner B to first train the additional GAN. So the last challenge that I will mention is that if you train a GAN on healthy controls for image-to-image translation, can you expect that it works for deceased subjects? So in general, you cannot assume that. So in this paper, they show that if they train a GAN for translation between FLAIR and T1, and they do the training on healthy controls, and then they apply it to brain tumor patients, the translation will actually remove the tumor, because the GAN has never seen any tumor. And if they instead train the GAN with images from brain tumor patients and apply it on healthy controls, it can instead add tumors. So when you train a GAN, you have to be a bit careful with what kind of data you use for training and the kind of data you use for the application. So we can see this as a bug or as a feature. So if you have, for example, a real T2 image from a deceased subject, then you can produce a healthy fake T1. And if you also have a real T1 from the deceased subject, you can then make a comparison between the healthy prediction and the real T1. And you can see the difference as some kind of abnormality. But then you can run into registration errors if you don't have a perfect registration. So you don't know if the difference is due to registration or due to a a real difference. So what you can do is to go from deceased through T1 to healthy fake T2, and then use the fact that cyclical GAN learns both transformations. So then we can transform it back to T1. And now all these three images are exactly aligned, so we don't need any registration. So there cannot be any registration error. Then we can compare the healthy fake T1 to the deceased through T1. So this is one example where they don't use exactly that approach, but they use something similar. So this is the original data. This is what they call reconstruction, or what I will call a healthy prediction. And then they can take the original minus the prediction, and the difference can be seen as an abnormality. Yes. So we have prepared a notebook that's available on the homepage that you can play with. And otherwise, I can try to answer your questions.
Hi, thanks for the talk. I was wondering, since you spoke about the memory consumption problems if you go to 3D, if you have considered invertible networks, if you're aware of this option and what you think about it?
No, I have not heard about this.
Okay, yeah, thanks.
Hi. Maybe I saw this wrong, but you showed an image of a GAN that took a T1 image and then produced a CT image and then back to a T1. And the CT image didn't seem to have any brain picture, yet in the synthetic T1, there was a brain again. Is that not a little worrying?
Well, it may seem like magic, but since I guess it relates to the receptive field of your convolutional network. So if it can see the full volume and it knows what the brain normally looks like in MR, you can still make sort of a guess what the brain should look like.
So basically, if I understand correctly, the GAN just made up a brain based on the shape of the skull.
Yeah, it learns what the brain normally looks like in an MR volume. And then it will generate a new brain that fits into your current volume.
But you also had a comparison then, the difference map.
Yeah.
And that didn't show much error. So basically, the invented brain magically matched the original very well.
It looks a bit like magic. But
it does. Thank you.
Thanks for your talk, Anders. I'm wondering, like, for the image-to-image translation stuff, isn't that sort of what UNet does? You put an image in, you get an image out, and usually it's segmentation. But do you have any intuition on why people don't use just UNets to generate modalities and that kind of thing, and where the gain is?
I'm not sure, but I think some researchers seems to use GANs for segmentation instead of UNet. So I think it's more like a matter of taste what you select.
Okay, well, thanks a lot. I think that's all the time we have for questions. So I'm just going to introduce the next speaker from here. Steffen would like to have his computer display his presentation. Is from the Centre for Advanced Imaging at the University of Queensland, from Brisbane, Australia. And he has a lot of experience working with ultra-high field MR and to try to relate the images that we take to some quantitative biological properties, and looking forward to the presentation. So,
yeah. Cool. Thank you for the kind introduction. So while we're waiting for the slides, thank you for organizing this cool workshop. So I really liked the sessions, and yeah, so thank you, Andrew and Ariel, for putting this together. Before I start, so who has heard about QSM? Oh, okay, a couple of people, that's cool. Good, I will still, I have a couple of slides to introduce what QSM is, just so we are all at the same page before we start doing some deep learning with this. Do you need anything else on my notebook? Do you see the signal? That's the backup slides, so we can. It should. Yeah, it doesn't get a signal. Yeah, I agree. Ah, that looks good. Okay, we got something. Yeah. Ah, fantastic. Cool. Thank you very much. Cool. Okay, so QSM, what is it? So basically, magnetic susceptibility measures the degree of magnetization of a material in a magnetic field. And it is computed based on the MRI signal phase of a gradient echo scan. And usually it has quite an elaborate pipeline to get this information out. So in the end, we have this quantitative susceptibility map, and we start from complex data phase, and we have to unwrap the phase, we have to background field to correct the phase because there are external sources that overlay this signal, and then we need to solve an ill-post inverse problem to get this QSM. And what is it used for? So it's quite sensitive to all biometals, microbleeds, calcifications. It can help us with exploring contrast agent biodistributions, and it can also be useful in exploring demyelination processes. So here we see a simple example where QSM could be useful. So there's an active lesion, so a bleeding is in positive, so in white, a positive susceptibility, and there's a calcified, or there are calcified lesions in here which show up as negative susceptibilities. And if we look as a comparison, the T2 star weighting, weighted image doesn't show it that clearly. Also potentially quite useful in multiple sclerosis because there we know iron accumulates after demyelination in microglia, and also people have observed slow iron depletion in normal appearing white matter. So QSM is quite useful, but how do we get it? So this is a simple toy example. So we have magnetic susceptibility distribution, so a little magnetic source that we put in the magnetic field. And what we get is, when we apply a magnetic field, is we get this magnetic field perturbation. So this looks like this dipolar pattern. And it's basically our susceptibility source disturbs the magnetic field. And when we measure it in MRI, we get something that looks like this. And it's the wrapped MRI phase, because the phase we measure between minus pi and pi. So when we hit the boundaries of pi, then we wrap back to minus pi, for example. So that's why you see this wrapping, and it's not a continuous function as we see it, as it is in a real physical way. So the first thing we have to do is we have to unwrap it, and then we have to solve this inverse field to source problem. And now you can think of this problem as, if we know our source distribution, we can simply compute the forward model, because this, we know the relationship. So we just know if we have a source, there is a dipole kernel that describes that. So I will show that in a minute. And in order to estimate which source caused this field perturbation, we currently use iterative procedures. So we simply assume a source, we propagate this forward, we compute the difference, and then we minimize this in the least square sense. So that's our current procedures. And we call this inverse problem, but it's computationally quite expensive and has its problems to solve it. So that's why we thought, let's use deep learning for this, because it can approximate any function, and we call that deep QSM. And how did we do this? So we did not use, I think this is probably one of the differences to the talks we already saw today. We did not use measured data to do this. We actually simulated data, because the problem is we can describe that. So what we did is we started simulating some simple shapes. So similar to the toy example I just showed you, just a couple more, because it doesn't like background. So if most of the image is background, the network will not learn. So we put a lot of these structures in there. Then we know our dipole kernel. So this is the mathematical description of this convolutional, or of this dipole kernel that we convolve our data with. And then we get our simulated magnetic field perturbation. And then what we did is we simulated lots of examples of these. And all of these images are three-dimensional, because as you can see, it's X, Y, Z. So this is a three-dimensional problem, and you cannot solve it on a single slice, because you miss crucial information from the other slices. So all of these images are 3D blocks. And then we generated 91,600 of these blocks. And in the beginning, I didn't know how much data I need to train. So I simply generated as much data as I could, and then my disk allocation was full. So that's how I ended up with 1,600. So that's one way of finding a hyperparameter. And then we simply trained it. So then we said, okay, this is my input and this is my target. And we trained a UNet to do this. And how does it perform? So now we simply simulated new data. We convolved it again, got the phase. And then here I have two established toolkits, STI, ALSQR. This is an iterative procedure, and MEDI. And these, I would say, are the two of the state-of-the-art methods that are very good and accepted in the community. And then on the right side, we see deep QSM. So these are the outputs when I put this phase in these toolkits. So we see that deep QSM generates something quite close to the simulation that we put in. When we look at the difference to simulation, it's good to see that all of them make some mistakes. So we see there are some, for example, here at some edges where the network is wrong. But it's also good because it shows that we probably don't overfit to the data. So we haven't learned the data. And also it's an ill-posed problem. So you would expect that there is no perfect solution to this problem, because if you want to solve it perfectly, you need multiple orientations. This is called Cosmos. And then you can solve the problem analytically. And then you don't have this ill-posedness. But here we have only one orientation, so there has to be some error. And then, remember, we just trained on shapes. So we thought, what happens if we use a real brain data set that was actually measured? And this is the phase of this brain. It's already background field corrected. And when we use STI, MEDI, and deep QSM, we see we get quite similar results. However, there are some differences. For example, MEDI, in this case, I didn't have a very high regularization of this model term, so we get a lot of streaking. So MEDI in this case took the data more serious than its model, and then that's why it ended up with some streaking artifacts. So basically these dark spots here, and you see it later also a bit more clearer. So we call these are artifacts, and these are due to this ill-posedness of this problem. And STI is quite similar to what we get in deep QSM. And then to investigate these effects a little bit more, we scanned 27 subjects. And now here we see a group result of a group template. So I simply averaged all of these 27 subjects so I can look at the artifacts on a group level. And what we see here is, especially in this coronal plane, we see these artifacts quite nicely. So these black streaks, this is what we call the streaking artifact, and also here you see it quite nicely. And even STI suite, where we didn't see it in the single subject, even here I think I can see some streaking. But when we look at deep QSM, there is almost no artifact there. And it seems that the network found a very good regularization for this problem, for this ill-posed problem. But we also see that we lose some contrast. So in comparison to these two others, we have we have a lot less contrast. And this could be that the network is over-regularizing there. That could well be. And now I want to switch to the demo. So theoretically, if you open this demo, you should get in Google Colab, as we already saw today. And then here I have to make a disclaimer. So the code I'm showing today is not the original DeepQSM source code that we used in the paper. So I wrote it newly for the OHBM educational course because actually a lot of the code complexity is reading in the data, shaping the data, and running it on a GPU cluster. And all of this is not really necessary for here. So I just wrote it from scratch to make it more easy to understand what we're doing. So the first thing I'm doing here is I'm mounting my Google Drive so I can actually store my information across sessions. And I created a folder in here, DLQSM, so this should happen also in your Google Drive, and then you can look at the data later. And then if you're following this live, you need to uncomment these two lines where we have to download the reconstruction challenge data. So there was 2016, the QSM community came together and said, we want to compare these different QSM algorithms, and they came up with a reconstruction challenge. So we're using this data to test our trained network. And then this is just a function to plot a three-dimensional data set. And then we are looking here at the reconstruction challenge data. So let's have a look how this looks like. So here we see, this is the face, tissue face. So you see it's blurry and it doesn't really look like a quantitative susceptibility map. And here we see, I called it gold standard QSM. So this is a multi-orientation reconstruction. So we actually can solve that problem analytically, but of course clinically that's not feasible. You can't rotate your patients in the scanner easily. So that's why I call it gold standard QSM, and also it's not perfect, so we know also this model has its issues, but yeah, we should see something like these bright structures in the center of the brain, and you see it's a lot less blurry, and this dipole effect is removed. So that's what we're aiming for. Now, let's simulate data. So this is a function that I also simplified from what we used in the paper. So here I'm just using some rectangular shapes because they're much faster to generate. And when we look at them, we see them here. So it's a three-dimensional block, 320 by 320 by 320. And I have 800 rectangular shapes in there, and then they're three-dimensional. And then this is how this looks like. And now I need to apply my dipole kernel. So this is this dipole kernel you saw on the slide. So 1 over 3 minus this dipole formula. And when we generate this dipole and look at it, we can see how it looks like. So this is the dipole effect in Fourier space. So this is basically how we filter our data. And this is the effect we want to undo. So now I'm computing the forward convolution of my simulated data to get this artificial phase data. And that takes a while. Yep, so basically I'm going in K-space, and in K-space, in Fourier space, and in Fourier space, I'm multiplying with the dipole kernel because convolution in image space is a multiplication in Fourier space. And then we can look at this. So this is the simulation. So this is, again, this is the output after convolving with the dipole kernel. So you see here I had, so here below you see the input, these rectangular shapes. This is our simulated ground truth, and here we see the convolution, or the output of the convolution. You see you have all these blurry things, and things are, it's hard to distinguish. So you couldn't say that this thing was black or had this value because of this, this effect. So now we are training a model to undo this effect. So the first thing we have to do is, and this is again similar to what we already saw in the other presentations, on the GPU we don't have that much memory. So what I chose here is I take patches, three-dimensional patches from this big data set. And here I generate 500 patches, and I make them 64 by 64 by 64. So let's have a look at this. This should be quite fast. And then we can look at patch index 44. And here we see this is the ground truth, and this is after convolution. And this is what we will use for training. So the input will be this blurred pattern, and the output will be, or the target will be this ground truth. Okay. Then I thought for this tutorial I used the newest TensorFlow because I wanted to play with some of the new features. So this is 2.00 alpha 0. So theoretically, you should be able to run this also in a few more weeks when the final version comes out. And just testing that my GPU is here. So I'm using in a runtime with GPU. And what I'm defining here is, so we saw today already how we build up these units. And I'm lazy and I don't like copy and paste, so I copied from this example. There's this pix-to-pix example from Google. This one, yes. So tutorial is generative and pix-to-pix, and they have a very nice way of generating a unit where you define the downsample function first, you have the initializer, and then you add a convolutional 3D layer. Then you can decide if you want batch normalization or not, and then you apply a leaky ReLU unit. So this is our downsample, or one downsample block. Then I'm testing that this works and get everything in terms of shapes right. So I have a 64 cubed volume, and then the first dimension for this TensorFlow model is the batches, so how many examples I show per training step. And the last dimension are the channels. So you can, currently I only have one channel, but if you have multimodal data, then you could have multiple channels. Then I'm testing this downsample part. So I'm giving it one of my input data, and I'm checking that I get the right dimensions out. So it downsamples from 64 to 32. And I made 8 filters, so I see that the filters are now where I'm expecting them, and I had one example. So that all works out. Then I have an upsample part. The upsample part, the only difference really is that I have not a 3D convolution, I have a convolutional 3D transpose. And I also apply dropout on the upsample part and only in the center of the unit. And also then there's a ReLU. So let's execute this. And then if it's not tested, it is broken. So let's test it as well. Yes, okay, so that works. 64 by 64, and then we get 8 filters. So we can upsample again. So I gave it the downsampled result to test if I can get back my 64 shapes. Okay, so this all looks good. Then I tried to get a model that trains while I'm speaking, and I couldn't get anything to work with training times less than an hour. I think the problem is that it's a 3D data set, and it takes a lot of time just to train. With a 2D data set, I think it could have worked, but 3D I just couldn't get to work. So I trained it with about 500 epochs, so it takes a while, and then I'm saving the data sets out every 20 minutes so that in case my notebook session expires, it still runs. Or I can still look at the results later. Then here I'm defining the architecture, so I'm parameterize the filter base, so how many filters I want, the kernel size, and then I'm basically downsampling until I'm at the base of the unit, and then I'm creating an upstack where I'm upsampling again, and the initializer, the last layer of the network, and then the concatenation layer. I'm defining the inputs. So here's an important thing because I want this thing to be fully convolutional. So if I wouldn't put none here, but the actual input size, then it wouldn't be able to then Keras complaints. So this gives me the possibility to train on 64 by 64 by 64, but then apply it to 160 or 320 if I want to. Yes, then I'm setting up the skip connections and also for upsampling, and then I created this model. That's basically the unit. And then I'm compiling it with the mean squared error and an optimizer. I used Atom. And then here we can look at this model. So we see the input, then we see our sequential, where we are going down, then our concatenations, and then we're going back up again. And we have 3.5 million parameters. So let's quickly test, what does the network do? So now this is defined, right? It's initialized, but we haven't trained it. So what is the output when we don't train it, just as out of curiosity? So this is the input, this blurry thing. This is the susceptibility distribution that we want. And when we give this as an input to the network, we get this as an output. So that's good. So we're not getting anything nonsense, and it also doesn't give us back the input. So it's just giving us a blank thing. And then we can train. So I will skip the training now. And I will load a checkpoint that I created beforehand. And then we can look at what the training is. So I trained it for 500 epochs, and we see the error goes pretty quickly goes down, and then it's on a downward trend for a few epochs. And then the first question, did we actually learn the training data? So just as a sanity check, so I'm giving it one of the training data sets again. So it has already seen this during training. This is the input, this is what it should predict, and this is what it predicts. So we see that the network is still not perfect. So even after training 500 epochs, it has an overfit to the data, so it's not reproducing exactly the training data. But we also see that it's still doing some mistakes. But when we consider that this was the input and it recovered this, that's not too bad. And also we have to think about it, it is an ill-posed problem, so there is no perfect solution, or it shouldn't produce the perfect solution. Then the question is, what happens, so this was on one patch, what happens if we give it now the whole data set, the big 320 data that I generated in the beginning. So just to see, this is 320 cubed, this is the ground truth that I'm expecting, and this is what the model gives me. So here we see that in some areas it is pretty off, but we also see that it recovered some of the details quite nicely. And can it work on brain data? So this is where I was hoping that it works a little bit better. And here we see the input, we see what we ideally would like to have, and this is where we currently are. So we are on the way to the ground truth, but we are not yet there. And yeah, but if we compare that, it's a lot less blurry, and we start to see the right contrast that we would expect for QSM. Yeah, and then I just added another model, a slightly simpler model. So if you want to run that, and then you can, as a last thing, I just have the influence of the hyperparameter. So you can play with this a little bit, and then we can compare the two models and see that in this case, the model one I showed you first, because it was a much more complex model, has a better error rate than the second model that was a much simpler model. So you can play with this. And then I'm going back to my slides, and that's just as a summary. Yes, so it learned to basically take this input, produce this output, and it's quite close to ground truth. But yeah, in order to get better, we need a lot more examples and a lot longer training time. And yes, and also on brain, we see it did get closer to the ground truth, but it's not perfect yet. And then I wanted to present another thing on top of this. So now we only solved the inverse problem of QSM. So the next thing is the background field correction. And we used a very similar approach to what we already showed with deep QSM. We called it SharkNet. And what are we doing there? So here we're not simulating susceptibility distributions per se. So we're actually simulating background sources. So this is a frame around the brain, and we simulate sources that are outside of the brain, and that they generate fields that look like these background fields that we see in QSM. And then we overlay them on a synthetic brain, and this is what the input would be to the network. So the question was, can we also remove this background field with the same technique? So we, again, simulated 1,000 source configurations. We got 1,000 simulated background fields, and we overlaid that on these synthetic brains. Then, again, these are all three-dimensional patches. So we cut 1,000 crops per 1,000 simulations. So we ended up with 100,000 3D patches. And then we thought, okay, let's test that. So we have this synthetic ground truth brain. This is a simulated background field that we overlaid on top of this. And we see that it's very hard to see the details below. So you see it's really, this background field is messing with us, and we couldn't use this to directly invert the problem. So we need to remove that first. And this is SharkNet, so this is the output of the network. We can, when we compare this to the ground truth, we see we are quite close. We managed to remove this low frequency background field. And when we compare to established methods, so sharp, resharp, and vsharp are all established methods. And we see, for example, sharp filters the image quite a lot. So for example, here you see a small dark band that actually is true and that should be there, because also structures inside the brain cause low frequency fields, so we cannot just spatially filter it. But yeah, with sharp, we lose this information, but resharp and vsharp are a little bit more sophisticated, or extensions to sharp, and they keep this information. Since this is simulated data, we actually get real error measures. So when we look at SharkNet versus ground truth, we see the errors are quite low compared to the established methods, which is very nice and good to see. And we also tested it in a real in vivo case. So this is measured data where we don't have grounded truth, and this is the real background field of a real volunteer. And we see here most of the background field comes from the sinus areas and then affects this part of the brain, also the temporal lobes. We have lots of this background field. And we see that all methods manage to correct this background field. And then, of course, it's hard to say what's true, but we could say from our simulations that we saw that sharp is removing a little bit too much. So we see we have some of these dark structures that we kept here, but that are gone here. And yeah, that's this. And I think with this, I thank you for your attention, and I'm looking forward to questions. Thank you.
Hey. You mentioned overfitting several times, but if you're using simulated data, and I don't recall you mentioning any noise, what's the problem of overfitting in that context?
It shouldn't be a problem. So yes, that's why we chose simulated data, because yeah, we would see it if we overfit on the simulated data. And then if it doesn't work on brain, it would be quite obvious. And we haven't trained with noise, but that is a idea that we should do.
Why did you not add noise?
Why we didn't add noise? Just because it was working without noise, so we didn't bother with it. But it would make a lot of sense.
Overfitting is usually overfitting to the noise. So I'm wondering if you can even have overfitting if there's no noise.
That is true. Yes, well, you could. I guess you could overfit to, if you don't add noise, you could probably learn the simulated structures.
Yeah, that would have been my next question. Does it matter? Have you checked if it matters what kind of shapes you train on?
Not really. Yes, exactly. So we checked, we played with this a lot, finding an optimal training case, and it seems to work with almost everything. So that's the funny thing. Where it doesn't work is if you have mainly background, then the network learns to predict background, because that minimizes the error nicely. So the image has to be full with stuff, and ideally I think the spatial frequencies have to match what we see in the brain. So I guess you could even just simulate random noise and train on that, if that has enough structure for the problem.
Yeah, reflect. But your geometric structures don't really match up with anything you see in the brain, right?
Well, when you look at the spatial frequencies, it does, because we have edges and we have things. So from this perspective, it does match. So we represent structure. So I don't think that if you have something that's completely unstructured, it wouldn't be able to learn that. But I think there's a lot of room for improvement there. So if you come up with a better way of simulating data that represents brain data on a spatial frequency level, I think that would be quite nice to optimize the training. Yeah.
Thank you.
Okay, thank you. Any other questions? If not, I guess we're moving on.
right, thanks again, Steffen. And we're going to take a quick break and come back in 20 minutes at 3:05. Okay, everyone, welcome back to the last session of the Deep Learning for Human Brain Mapping educational course of 2019. And our next speaker is Jacob Wassertal, who is a PhD student at DKFZ, and he's going to talk to us about his work using deep learning to do tractography and diffusion data. So take it away, Jacob.
Thank you very much. All right, so I'm from the German Cancer Research Center in Heidelberg, and today I'm going to show you how we can use deep learning to reconstruct white matter bundles. So what is this about? So basically, if you have a diffusion-weighted image of the brain, what we want to do is reconstruct the different fiber bundles within the white matter of the human brain, for example, the corticospinal tract. And so how would we normally approach that? So the normal approach normally would be, first we fit some model to our diffusion image. For example, some tensor model or some more sophisticated constraint spherical deconvolution model to model the signal in each voxel. So for each voxel, then we get some fiber orientation distributions. And then we can run whole brain tractography, giving us millions of fibers for the entire brain. And then if we are interested in specific anatomical bundles like the corticospinal tract, we have to use some ways to extract this bundle from this millions of fibers. And you could do it manually, or there are some automatic approaches using clustering or using some atlases and stuff like that. And now the question is, can we use deep learning for that, and can we gain anything from that? The basic approach we were using is using a UNet. So I guess most of you have seen a UNet. Just a quick explanation. So the UNet is maybe the most used model for semantic segmentation in medical images. It's a convolutional neural network. And so what it's doing, so all those blue layers here, those are convolutional layers. And basically here we have a downsampling path where it reduces the spatial dimension. So, and at the same time, it's increasing the number of filters. So here in the beginning, we still have the entire image size. And then we come down here. Spatially, the image are getting smaller and smaller, but we gain more and more filters, and those filters encode some semantic information that the model is learning about the image. And then here again, we have an upsampling path. So down here, that's basically what you would do if you would want to classify images. So if you want to classify dogs and cats, you would just have this downsampling path basically. And down here you have a lot of semantic information about your image, and then you can just add a fully connected layer which tells you is it a dog or is it a cat. But in our case, we want to have a label basically for each voxel and not one label for the entire image. And therefore, we have this upsampling path where we gain spatial information. And though here, we are upsampling the spatial dimension back to the original full image resolution. And then here we have basically one label for each voxel in the image. And then in here, that's basically some skip connections, so the information can also go this way to give the network the ability to learn some more detailed, more spatial details, which might get lost if we go this way down here and then up again. So that's a very, very rough explanation of what the UNet is doing, and it's allowing us for an input image to learn a segmentation. So the very classic example would be learn a segmentation of a brain tumor, for example. So you input your brain image, and then you want to learn the segmentation of the tumor, for example. So that what it would be used for example. And now in our case, what we want to do, we want to learn in a first step a segmentation of the different bundles in the brain, for example, the corticospinal tract. So the toolkit we developed for that, we call it TractSeg, and it's based on the UNet, so that's the very core of it. And so how do we use it for segmenting tracts? So first, the input is the following. So we have our diffusion-weighted image. Then we fit a model, in our case, a constrained spherical deconvolution, to estimate the fiber orientation distribution in each voxel. And then we extract the main peak of that, and that basically gives us for each voxel the main orientations of the fibers at that voxel. And what we have in here is basically 9 channels telling us the 3 main fiber populations. And now each of those peaks is just a vector in 3D space encoded by 3 numbers. And if we stack those, we basically have 9 channels. So the input to our network is just our image with 9 channels encoding those 3 peaks in each voxel. So that's the input to our network, and then the output, what it's learning is just a binary segmentation for those different bundles. And in this case, we trained it for 72 different bundles to segment all of them at the same time. Therefore, we have 72 different output channels, one for each bundle. So now one important question, of course, how to deal with the 3D data. So one approach would be just to process it slice by slice. So you would input a 2D slice to the UNet, and you would give it all 2D slices of your entire brain, and then in the end you can stack them back together and you got your 3D segmentation. Or what you could also do is use a 3D UNet. So you could use 3D convolutions, and then you could input an entire 3D volume, and it would output a 3D segmentation. Or there's a third approach, which is often called 2.5D. So basically it's the 2D approach, but we are sampling those 2D slices from several orientations. And then basically we can average the result, giving us kind of an ensemble. And generally speaking, you would assume that the 3D model is always working best, but it turns out that for some medical problems, 2D actually works a little bit better. And so from my experience and the experience in our lab, sometimes 2D works better and sometimes 3D works better. There's not a huge difference, but a small difference, and you you can't really tell that one of those two is always the better option. Though in this case of tract segmentation, it turned out that the 2D approach worked better, and then we actually extended this to this 2.5D approach, and having this ensemble of those 3 models gives a small performance increase. So that's the way we went. Now another question, of course, is how to tweak all the different parameters of the UNet, and what about all those fancy extensions you hear about in the last years. And there have been quite a few papers proposing a lot of different modifications to the UNet. For example, squeeze and excitation layers, which have been quite successful, or which were the winning entry of the ImageNet challenge 1 or 2 years ago, I guess. And there were some attention mechanisms people built into that, and they're basically showing that for their data set, they gained some performance by that. But actually, in our lab, when we were using that, it turned out that often when we re-implemented those, we didn't really gain anything on our data sets. It turned out we didn't really gain a lot from adding all those fancy adaptations to the UNet. And it turned out that the UNet itself in its standard configuration is actually really powerful. And if you tune the hyperparameters of the standard UNet, you can actually gain really good results. However, getting those normal or the standard hyperparameters right is not that easy. So what we actually did is we built something we call the NN UNet. The NN stands for no new UNet because it's not something adapted. It's just optimizing the standard UNet basically. And so what is it, the NN UNet? So that was the entry of my lab and especially my colleague Fabian Isensee to the MICAI Decathlon last year. And so basically the challenge was to have one model that works best on a lot of different data sets. And it turned out that with this model that is using a normal UNet, but really trying to optimize the parameters like the learning rate decay, your loss function, how you weight your loss function, and all of those different parameters you can tune. If you really optimize those, you can actually get the best performance on a lot of different data sets. We can see here in this plot, like the red dot, so that's the NN UNet, and the blue dots are those other competing entries. And it turns out that actually this normal UNet really works best if you look at a lot of different data sets and not overfit on one data set by really trying to optimize your hyperparameters for only one data set. And yeah, it's openly available, this NN UNet, and it's quite nice, you can just transform your input data or your data set to the right format, which is required from the NN UNet, give it to the NN UNet, and it has some automatic heuristics to use the right parameters for the UNet, and then it will automatically train it, and you will get some pretty good results. That's just a side note on when you work on semantic segmentation with the UNet, don't waste too much time trying to add some fancy stuff. Probably you are more overfitting on your data set instead of improving the UNet on a lot of different data sets. Now coming back to the actual thing I was talking about, to the tracked segmentation. So I've shown you the approach I'm using. And now the question is, how do we get training data for that? So what we did, we took 105 subjects from the Human Connectome project, which has a really nice quality. And then we took a semi-automatic approach to delineate those 72 different bundles. So we did a whole brain tractography, and then we used different tools like tracked query and some clustering and atlas approaches to extract those different bundles. And that did a lot of the job, but still was not perfect yet, though we did quite a bit of manual quality control and cleaning up results where it was not perfect yet. And that gave us the data set we used to train our model. And one important thing you can do is data augmentation. So we augmented our data quite heavily. We did some elastic deformations, we did displacements, we zoomed our image, we added Gaussian blurring and Gaussian noise. And you have two examples where we can basically see what's happening. So on the left-hand side, we have the image. So that's one channel of this peak image. Therefore, you have those crazy-looking gray values. But on the left-hand side, you can see the image without data augmentation, and on the right-hand side with data augmentation. So the data augmentation is done on the fly and basically randomly. So we randomly choose how much Gaussian blurring we are doing, randomly choose how much Gaussian noise we are doing, and so on. And on the first one, we can, for example, see if we look at the corpus callosum on the left and we compare it with the one on the right, we can see how the elastic deformation deformed it quite a bit. But still, we keep it in a range where it's still looking kind of realistic, the deformed image. And the nice thing about that is, so if you add this data augmentation, And when you apply your network to a new data set, you don't have to register it to a predefined atlas space, at least not perfectly, because it learned to work with images that are displaced. So the only thing we need that it's having the same left-right, front-back orientation, but it does not have to be perfectly aligned, so you don't have to care that your registration is working really well. So now let's have a look at some results. Though in green here we can see the corticospinal tract from our training data, and in red we can see the results from our tool. And we can see that it actually managed pretty well to learn to recognize the shape of the corticospinal tract. So when we look at it in terms of numbers, we compared it to a lot of different toolkits, some using atlases, others using clustering. It turns out that we also perform really well. So the first one, that's our toolkit, and we compared it on HTTP quality data, then on HTTP quality data downsampled to a lower resolution. That's what we see in the middle in red. If you downsample it and remove a lot of the gradient orientations. And then on the right-hand side, we also simulated brains using a software phantom. That's what we see on the right-hand side. And for all of those, we perform really well when looking at the DICE score. So a DICE score of 1 would be perfect segmentation. And we are doing quite well. So the next question is, can we do more than just segmentation? Now we have a binary segmentation, that's nice, but for quite a few applications, you want to have those streamlines. So what we did is binary segmentation. Then what you could also easily do is, instead of learning a binary segmentation, you could learn a density map. If you input the density as in your training data, or what you could do, you could learn a vector field actually. So basically for each voxel, now you are learning one vector. For the binary segmentation, as I've shown before, we basically need one channel for each bundle. So we have 72 times 1 channels. From the density map, it's the same. And then when we learn a vector field, we need basically 3 channels, because again, it's one vector in 3D space. So we have 72 bundles, and for each we need 3 channels. So it gives us 216 channels. That's the output of the UNet. So how do we train that? To learn those peaks, you could just use a mean squared error. It turns out that works okay, but not so well. So what works better is to use an angular error. And that gave really nice results. So just as a loss function, use the angle error between the ground truth peaks and the peak that we are learning. So now we can put all of that together and to generate quite nice bundle-specific tractograms. So here in the middle, we have our binary segmentation of the tract. Then on the top, we have those vector fields for each bundle. And then on the bottom, we also, in another network, learn the region where the fibers of each bundle are supposed to start and end. And then we can just use a tracking algorithm to track on those vector fields and filter the streamlines, or filter all streamlines that leave the tracked binary mask and do not end in those start and end regions we have learned. And that gives us really nice reconstructions of those tracts. Let's have a look at some results. So here, for one subject, if we would try to manually reconstruct the corticospinal tract, you first have to run your whole brain tractography. Depending on what parameters you use, it takes quite a while, and then you have to manually draw a lot of regions of interest to remove all the fibers you don't want. If you use our algorithm, it runs a lot faster because you don't have to use this whole brain tracking, and it gives you quite nice results. And as we've seen here, so this is on a subject with schizophrenia. And so we have only trained it on those HTTP data with data augmentation. And as we can see here, it's really nice that it really transfers or generalizes to other data sets without having to do anything else. So apparently the input, we are inputting those peaks, and those are giving us a nice abstraction, making it kind of independent from the input sequence, and at the same time using this data augmentation also helps to make it transfer well between different data sets. Here you have some more results. So this is a subject with Alzheimer. And then we can see this subject is having really big ventricles. And our model also learned quite well to deal with those big ventricles. So it managed, it reconstructed the corticospinal tract around those ventricles. And on the right-hand side, when looking at the optic radiation, you can see, despite of this really, really large ventricle, it managed to find its way around there. So it's really interesting that despite being trained only on healthy subject, it works well even with distorted brain anatomy. So this toolkit called TrackSec is openly available on GitHub. You can just use it with Python, and it is pre-trained. Our data set for the training is also openly available. And now let's have a look and try it out. So we have a IPython notebook for that. Let's see. It's on, here we go. Okay. So this is gonna be a bit more challenging. Okay. Let's have a look. Okay, that looks better. Okay, perfect. So on the website from the educational session here, there should be the link to this notebook if you want to follow along. And that's what it looks like if we open it. Maybe we make this a little bit larger. Okay, I hope everybody can read it now. So here the beginning is quite boring. It's just installing TrackSec. It's just pip install and then basically our GitHub URL. So that's quite easy. And then here we are downloading some training data. Oh, some example data. Sorry, it's just some examples so I can show you something. Yeah, and here I'm importing some utilities so we can have a look at the results. So I already ran those, so I'm not running those at the moment. And now if we look at the folder example files, nice. So we have some example files. We have a diffusion image, so that's just a plain diffusion weighted image with B-wells and BVECs, a brain mask. And now we need those peaks. I already extracted those using matrix. If you did not do so beforehand, you can just run TrackSec and calling the input raw diffusion input or using the argument raw diffusion input, and it will automatically call matrix in the background and calculate those peaks for you. Yeah, but there, yeah, you need matrix to be installed for that. So now we already did that. I did that beforehand, so I have this peaks file here. And what happened here? Here we go. And now if we look at this peak file in a viewer like MRView, it would look something similar to this picture, just to give you an impression what it kind of looks like. So you have those vectors. It's a little bit, the quality is not so good, unfortunately, here. It's giving you basically this vectors for each voxel. So that's the input, and now we can just call tractsec. So you just call tractsec, input the peaks, input or specify an output folder, and tell him that you want tract segmentations. And if we run this, it will first download the pre-trained model. So this will be automatically downloaded. This will only be needed when you do it the first time. After that, it will already be on your computer. And then it will process the file. And if you have a GPU with CUDA installed, then it will use the GPU and be really fast. If you don't have one, it will use the CPU and take a bit longer. So with a GPU, it normally takes less than a minute. And with CPU, it might take like 5 minutes, still quite reasonable. And now we can see it's processing those 3 images, or the image along 3 orientations. That's the approach I've shown at the beginning, this 2.5D approach. Therefore, we see those 3 progress bars, which don't look so nice on Colab, unfortunately. So normally that would show a nice progress bar. And now it's merging all of those 3 things together, and now we are basically done. And now if you look into the output folder, there's a subfolder now, bundle segmentations. And if we run this again, we can see now a binary mask for all of those 72 bundles. And now if we look at this binary mask in 3D space, it looks like this actually. Let's see if let's run this again. So we are plotting the binary mask, and I'm plotting also the brain mask with a reduced opacity. And then we can see here we get a nice segmentation of the corticospinal tract. And we can have a look at something else, for example, the optic radiation. And let's see, that also should give us some nice binary mask. Hopefully. It's taking a little bit longer, unfortunately. Here we go. Yeah, so here we can see the segmentation of the optic radiation, basically. All right, so now we have our binary mask. What we also want for our pipeline are the segmentations of the start and end region. So what we can do to get those, we just also call tractsec with the input peak, the output folder, and now we want output type ending segmentation. And yeah, so then if we run this, we will get those ending segmentations. I will not run it again because we are a little bit short of time. And now we can have a look at them. So it's giving you one output for the beginning of the bundle, one of the ending. And now if we merge those 2 and plot it, it looks like this for the corticospinal tract. So basically it's supposed to start down here and move up here and end in the precentral gyrus. Okay, and now we come to the 3rd step, learning this vector field. We call it a tract orientation map. And to learn this vector field, we just change the output type to Tom for tract orientation map. And again, it will we will run it. It takes approximately the same time as before. So if you have a GPU, quite fast, around a minute, something like that. And then if we look at the output peaks, it will look something similar like this. So here for one slice of the corticospinal tract, we get this vector field telling us the main orientation of the fibers of the corticospinal tract at this specific voxel. And so now the next step is to do tracking. Now we can use all of that to create streamlines. So there is another binary that comes with TrackSec. It's called tracking. And to tracking, again, you input your peaks. And you input the output folder where all of the other previous steps, the output of the previous steps is. So this is supposed to be the same output folder we have specified before. And yeah, and then, well, test, you can leave that out. That is just for testing it on only on 3 bundles. Normally it will run it for all bundles. And number of fibers, normally it's 2,000. Here I reduced it to 1,000 to make it a bit faster. And then we can have a look at the results here. So if we plot those, it looks the following. So here what we see again is the brain mask in gray, and here in red with the low opacity is the start and end region of the corticospinal tract. And now we see the streamlines. Let's zoom a little bit in. Of the corticos that were created for the corticospinal tract. And we can see it gives us yeah, a nice bundle-specific tractogram. And yeah, this is generated for all of those 72 different bundles. And this tracking command also is running. It doesn't take too long because it's only generating 2,000 streamlines and not several millions. Yeah, and now if you look at your output folder, you will see, yeah, there's this folder containing the bundle segmentations, the folder containing the ending segmentations, the folder containing the tract orientation maps, and the folder containing the trackings on the tract orientation maps. All right, so that's a very basic way how to use TrackSec to create those bundle-specific tractograms. Yeah, so it's quite fast to run, which is nice, and as I've shown in my presentation in the beginning, it really generalizes well to a lot of different data sets. Yeah, even so it was only trained on HTTP data with data augmentation. Depending on time, so there's the next part is just some information what you might want to consider if you use it on your data set. So as I mentioned before, it should be aligned, so left and right and front and back should be the same as the MNI space, and one easy way to ensure that is just rigidly register your image to MNI space. So it doesn't have to be a good registration, it just should, the overall image brain orientation just should match MNI space. And another thing that sometimes happens, depending on how you acquire your data set and how your gradient encoding is, for some data sets, when you look at the peaks you created with a matrix, you will see that they look kind of strange. So for example, here for the corpus callosum, the peaks go kind of this way, but they should go, well, along the corpus callosum. And that's because apparently sometimes the convention for how to encode those gradients is a little bit different. So you might have to flip them along the one axis, here in this case the x-axis. So just changing the sign on the x-axis. And then if you look at them again, you will see that they look just fine. So that's for most data that's not the case, but I came across a few where apparently the gradients are just encoded a little bit differently. Yeah, and just a maybe quick note on some advanced option you can use. So normally TrackSec will take your input image. So quite often your input image is a lower resolution than the HTTP data, then it will upsample it to HTTP resolution, that's 1.25 millimeters, and then it will process it and then downsample it again to your original image resolution. If you tell TrackSec this option super resolution, it will basically keep it in this 1.25 millimeter resolution where it was trained. So you can gain a higher resolution for your output than from your original image resolution. And it often gives a little bit better results if your image resolution is quite low. And then a final remark, so you can also use, when you do tracking, you can simply track on the vector field that is learned, but in some cases you might want to stay closer to your original signal. So what you could use the vector field that you learned for, so you could from original signal has like 3 peaks for each voxel, and now you could just select the peak which is closest to the one you learned. So basically you're just using the peak that is closest to the one you want to use. So for the corticospinal tract, so for example in this area where there's a lot of crossing fibers, you can just get rid of those that actually belong to a different bundle, and therefore also make tracking easier while still using like the peaks from the original signal. Let's see that. Somehow it's not connected anymore, but that doesn't really matter because we're almost at the end. One last thing, you can also use bedpost-X as your input. So if you already processed your data with bedpost-X and you don't want to process it again with matrix, you can just input the peaks from bedpost-X to save you some work. And if you look at tracks like --help, you will see a lot more documentation about all the details. So though much, that's all from me. Are there any questions?
Thank you very much for an interesting talk and this awesome software. I have a question about application of TrackSec to clinical data because I've tried to apply it to my data and it worked perfectly as long as I was using healthy volunteer data. But when I tried to move to clinical data, something went wrong. I admit that I have a very particular population of subjects. These are acute stroke subjects. So they have very abnormal diffusion characteristics. But I was surprised that even on the contralesional hemisphere, I was not able to detect, to segment tracts. And I'm wondering what could be, what might be the weak stages in your algorithm, at which stage it may break, and if you have any ideas how it could be fixed to deal with stroke patients.
So from my experience, so I tried it on a lot of different data sets I could find publicly available, and from my feeling it always failed somewhere in the pre-processing. So most of the times the gradients were flipped along one axis compared to the HCP data, and that was actually giving the problems most of the time, or not. The image not having the same left, right, front, back orientation as MNI space. That what I experienced most of the time actually gave the problems. So when everything was right, then actually for all the data sets where I tried it out, the results looked quite reasonable. Of course, if you have like major brain tumors, of course then the results might also be not so good. Right in the region of the brain tumor. But of course, if you're talking about brain tumors anyways, it's quite difficult to really say what's going on inside or quite close to a brain tumor. So that's hard to judge what's right and wrong there. Yeah.
Thank you.
Yeah, but along the same lines, I noticed you work at a cancer research center and you didn't show any of the images with the brain tumors. So what do you envisage as application of this software if it is not reliable in pathological cases and close to brain tumors?
So why I'm quite careful talking about brain tumors, I think is mainly because it's hard to say what's the ground truth around a brain tumor. So in a healthy brain, we are quite certain what the corticospinal, for example, is supposed to look like. But if it's distorted by a brain tumor, I think even for an expert, it's getting more difficult to judge. So from looking at results on brain tumors, they, looking at them, when our expert looked at them, they looked quite reasonable. But we didn't have a chance yet to really try to validate it using like intraoperative stimulation, stuff like that. And therefore, I'm rather careful talking about how well it works. So yeah, it looks quite well, but yeah, I can't say yet if that's really, it's not really validated yet properly. So another application then, I guess, is doing analysis in population without tumors, like what I've shown, like in schizophrenia, like if you have enlarged ventricles, those more homogeneous deformations compared to tumors, I think there the results are quite trustworthy. And there, if you want to analyze the different tracts, I think that's a possible application.
Thanks. It's going to be, again, along a similar sort of clinical lines. So you also predict or simulate other parameters coming from the tracks based on healthy populations. Have you used those, as was touched on earlier, as difference detectors or anomaly detectors in your clinical data? So you guess or predict what it's meant to look like based on the HCP data and then compare it, these density tractograms or what have you, to the clinical metrics to look where differences are?
So I'm not quite sure if I understood your question correctly.
So above and beyond predicting how the tracks look, so you've got a visual representation of your tract, you've also extended it to look at orientations, is that right? And
exactly,
look at density grams. So those are, again, simulated predictions of what you anticipate the value to be. And that's been derived from a healthy population. Have you quantitatively compared those in disease groups? Because you might see that you get greater divergence in the areas with pathology that deviate from the anticipated model.
No, we didn't evaluate those in deceased populations, no.
Okay, it might be interesting to.
Yeah, definitely, yeah.
All right, we're out of time. Thank you very much, Jacob, for that great talk. I'm amazed at how clear and great it was, given that you flew in an hour ago and were at a wedding last night. So hopefully I can do a good job too. Let's give another hand to Jacob. The next speaker is me, Andrew Doyle. I'm almost a PhD student at the Montreal Neurological Institute, and right now I'm doing a lot of signal processing on EEG data so that I can finally do some deep learning, and I have a poster on that, but that's not what I'm going to talk about today. I'm going to talk about looking inside the black box. Okay, and I get to go to the middle here because Ariel and I asked all of our speakers to prepare nice hands-on content, but I figured everyone's laptop would be dead by now, so I'm not going to do it. So, looking inside the black box. This is maybe one of the more dangerous talks I've ever given because it's like a little bit of a survey towards a bunch of different interpretability methods, and with deep learning, the black box is a little deeper, and you know, if you gaze long into an abyss, the abyss also gazes into you. And there's a couple reasons this is dangerous, but let's go in. So I was inspired to kind of develop this talk because last year at this same course, Professor Alexander Binder gave a fantastic lecture on his layer-wise relevance propagation method, which he is one of the main developers for. And I loved the talk, but didn't quite understand all of it. So I went back and read the papers and developed a sort of similar talk and then realized it was the same talk, so I changed it, and now it's hopefully unrecognizable to anyone who might have been at the course last year. So my approach is a little different to interpretability. I figure, why don't we just ask the AI directly how to interpret its predictions? So OpenAI recently released this text generation tool, and I asked it at talktotransformer.com, how can we interpret the prediction of deep neural networks? And this is what it spat out. Deep learning is not perfect, and there's nothing stopping us from improving the algorithm, but the current state in the marketplace implies that the machine learning is being more widely adopted than it has in the past. If you think about it, how important is it to get a computer to do all the computations while leaving the user and content completely unaware of what's going on? This is probably exactly the goal of deep learning. So this doesn't sound right to me. Maybe this was the wrong approach, and we should go to the literature and see what other scientists have done. I don't think the AI is ready yet, but you know, it's getting there. So quick review of GANs. I know we've heard a lot about them today. You start with this latent vector, Z, and we want to have a function G that generates some image, maybe from it. So you reshape it into a shape that kind of might resemble an image. Then we stack a bunch of layers, upsample, make it even bigger, add some skip connections, call it a ResNet, do some training. And this big GAN architecture is one of the state-of-the-art GANs for generating data. And this is supposedly a way we can do interpretability, is to generate the data from more or less scratch. So you can ask it to generate, by conditioning on the class label, a butterfly or a cheeseburger, pictures of islands and dogs, and sometimes it doesn't work too well if you haven't trained it enough, and you end up with dog ball here, who is neither dog nor ball. But you get some pretty nice samples out of it. And Big GAN discovered this trick. The latent space of Z, a lot of GAN papers draw from a uniform distribution, but Big GAN used a univariate Gaussian, which we heard Bliss talk about earlier. So I played around with their Colab notebook, which is linked to in this presentation, and generated a bunch of crocodiles and a bunch of chihuahuas. And to try to understand the similarities between them, I interpolated the Z vector between them and generated whatever that is in between. So I'm not sure we're gaining any understanding of how the black box works here, but maybe if you look kind of center left column, foxes are semantically related to chihuahuas and crocodiles. I'm not sure. But what they did discover and explore in the paper is that this truncation trick, if you truncate the Gaussian and don't sample as much from the tails and just sample closer to the mean, you end up with better quality chihuahuas and crocodiles, but less varied. So tried the interpolation here. The foxes are still there, so maybe we're gaining some understanding of biology. Links here. It's tons of fun and super easy to play with. Truncation trick is all you need to understand. So it's hard to evaluate the generator of GANs, whether or not it's doing the right thing. Because ideally, we want to be from the true distribution of the data, but the goal is to try to find it. So it's not super clear how to do this. And the ImageNet, cats and dogs and toasters and airplanes people came up with this inception score where you take some samples generated by your GAN and you pass them through InceptionNet, which is a state-of-the-art computer vision model, and you look at the probabilities of the labels. And so for good samples, you want low entropy in the predictions of the network because you want to be pretty sure of what the image is to say, oh, these are good images. But you also want high entropy in the prior distribution over the labels so that you generate varied samples. So this is one of the quantitative metrics, And if you put this dog through InceptionNet would probably guess like a human that it was a dog, but our friend Dogball here he has a little higher entropy because it's not clear what he is to anyone. So if you take the KL divergence, this is one of the ways to quantitatively evaluate this. Now the Fréchet inception distance is another way that also uses InceptionNet, but instead chops off the end and then puts a bunch of real images in, sees what the activations are at this layer where we've chopped it off, so it's not a classification yet. Does the same thing for generated images, and then you use the Fréchet distance between these two as a kind of quantitative measure of does my network see data the same way. So neither of these measures are really applicable to the kind of data that any of us deal with, because InceptionNet is a fantastic model for cats and dogs and toasters and chihuahuas and crocodiles, but it's not clear whether it's the best architecture for healthy controls versus autism in MRI image, or, we don't have that. We don't have a benchmark deep learning pre-trained network that we all compare against. And we have many, many image modalities. So it's a little bit harder, but we heard some ways that you can actually evaluate the generator easier from Bliss's talk earlier. So big GAN, high quality samples. The other really nice state-of-the-art GAN that came out last year is style GAN. And so the style GAN is really the first GAN that changes the architecture of the generator substantially. And normally on the left here, we've got sort of a standard setup where we start with a latent code and we stack a bunch of layers and make it bigger and end up with an image somehow. StyleGAN takes a bit of a different approach. So first there's this mapping network F. So you still have a latent space Z, but then you find a different intermediate latent space W, where hopefully this intermediate space is going to have linearly separable styles that are semantically meaningful and that actually encode the real features in your data that you care about. So also, there's another pathway in StyleGAN where they inject noise, like pure noise during training and testing, to model the parts of the image that aren't really semantically related to anything and just related to randomness. And so this is kind of what it might look like if the real features have this distribution where there's parts of it that there's no data for. So you might imagine that there are far fewer bald women than there are men. So the space of features in a face recognition data set would have very, very few women with shaved heads, let's say, and vice versa, there are comparatively fewer men with long hair. A normal GAN has this kind of mapping that doesn't take this into account at all, and it just tries to learn the best system, and it's uninterpretable in the end. StyleGAN tries to make this more related to the real features. And they really generate some fantastic results, even more impressive than the progressively growing GAN that some of the other speakers displayed. And you can see these are all completely synthetic examples. And on each row, you kind of have similar skin tones. And in the columns, you have other semantically meaningful things, like the left column has glasses, the middle column is a young boy, and the column to the right of that is a woman who's looking slightly off to the side. So there's really like a lot that can be modeled here. And so I found this interesting, and I used this technique to modify the smile, and I maybe let it run for a little too long, and it looks a little crazy, but you can also learn different semantically meaningful directions like gender, and if I get more male, I grow a beard, and if I get more female, I get longer hair and dimples, and I think I'm pretty as a female. And I tried this on Ariel too. And he looks pretty fabulous as a woman too. And you know, his hairline starts to recede as he gets more male. And you can see the difference between these two GIFs of the noise channel, where I've randomized it between each frame for me, but kept it static for Ariel. So the background is changing slightly, and the hair position, which is like, you know, not a semantic thing. It's like depends on how long I took to shave and that kind of thing, comb my hair. And I think this is more, we're starting to get a better intuition about the black box now. There is a weakness to the model though, and I tried this with age. I look pretty childish as a young boy, but there is no way I could ever grow such a glorious beard. So there are weaknesses to this style GAN. And I'm using this with some radiologists in my millennial side hustle outside of neuroscience to work on a big mammography data set to try to generate synthetic data to balance classes for some work that we're doing there, which is not finished, but it's still generating some compelling looking data. Can't show you the real images, but they look similar. So I'm not sure we got completely into the black box by using a model to generate data, but let's do a quick survey of the other stuff that's out there and the other ways to interpret predictions. So in terms of understanding the model, activation maximization is a technique you can use to find the input that for a deep learning network maximizes the prediction for one class. And I'm not sure what we're supposed to gain in terms of understanding and interpretability from these images. This is like the most representative dumbbell, cup, and Dalmatian for I think this is VGG 16. Not sure that this is useful, but you can use a similar technique, Deep Dream, to create some pretty psychedelic visualizations. And this is a surface of my T1 that I've put through Deep Dream, and all I can really tell is that I've got some demons in here. But again, you can go to the URL at the bottom here and generate these fairly easily yourself and play with the systems. So let's try to look at the filters now and visualize what's going on in the black box. The first layer is pretty easy. You can actually just plot the weights, and this is from the AlexNet paper that's come up a few times today. They used to, way back in the day in 2012, use big 11 by 11 convolutional filters for the first layer at least, and this is what the filters selected for when they slid across the input image. But if you go a little deeper, you start getting nonlinear combinations of the outputs of these, which very, very quickly become completely uninterpretable. And if we wanted to tell what does this layer do exactly, looking at the weights of these little 3 by 3 filters doesn't really help, especially because maybe there's 32 channels. If we can look at the activations, which gives us sort of a little bit more, but only for this single image that we've put in, and there's still a lot of them. Not sure that this is still a useful strategy. So deconvolution is a way to kind of visualize what each filter responds to most strongly over the whole input. And it's basically a collection of hacks to say, okay, let's say I had this activation for this filter layer. It's smaller than the input space though, because we've done max pooling. So let's save the location of where the maximum occurred so that we can propagate this back to the input. And we're still going to lose some information here, but if we do this over and over again, we can get all the way back to the input space. And so for these filters in layer 4, these patches got maximized, activated maximally, and deconvolution kind of on the left here produces this visualization of what the filter sees in these images to make them activate so strongly. So it turns out, though, that if you don't use max pooling in your network, this really doesn't work very well. And it's because of the way they dealt with the rectified linear unit, which are pretty ubiquitous in feed-forward convolutional networks like this. And so normally, when you're doing the forward pass of and trying to make a prediction, negative inputs get clamped to 0 by this ReLU. And in backpropagation, this is taken into account because you don't want to update weights that didn't create the signal. So when you get a negative gradient backwards in these yellow squares, you zero out where the ReLU was clamped to 0. But they didn't take this into account for the deconv net. They just did this backwards zeroing along with their max pooling thing. So this is just a combination of both the backpropagation strategy to go through the ReLU and the backwards deconv net approach. And this produces some nicer visualizations on a wider category of neural networks, and this is sort of the state of the art of how to visualize like what a filter sees in its receptive field when it gets activated strongly. And so you can see on the left here, the deconv are very blurry without the max pooling, but guided backpropagation seems to work. So the next way to kind of interpret predictions is to not try to look inside, but try to generate a heat map of what parts of the input were most strongly related to the prediction. And I hesitate to use the word caused because not a lot of this work is about causal inference, but I think that's the goal, is to try to get at causality, what parts of the input space cause the prediction. So a pretty good way to generate a heat map is to do occlusion testing. And it's very expensive, and you basically just take an image, you know this is a Pomeranian, and you artificially occlude every possible part of the image and see how the prediction changes. And the true label of Pomeranian is most certain whenever you don't put this square right over the Pomeranian's face, and then you're predicting dog. You know, it turns out dogs and balls are more semantically related than one might think. So, but this is definitely not how the brain does it, though. There's no demon in my head that's sliding around a gray square when I try to figure out what things are and what parts of objects are meaningful. The other way you can kind of pose this problem is using saliency. So, but this is now a little bit of a different question, and the question now is really, what if we change parts of the input space? How does that change the prediction? Which is maybe, hopefully, related to what causes the prediction, but it's not the same question. And we use this Jacobian matrix, which is in the shape of the input image, and this Jacobian matrix is really just the best linear approximation for the mapping that the network makes around the space of the data point that you're trying to explain. And so if you don't like live in this probability land where gradients are intuitive, it's important to draw this out. Backprop, the way it works is you calculate this loss function by going through your network, and then you take the gradient of your loss with respect to all the parameters of the network. And this is what you normally use derivatives for in this space. The saliency is very, very similar to this, except you're now taking the derivative of the output prediction, so there's no loss function anymore, with respect to the inputs. And you don't care about the weights now because we're not trying to train, but it's very similar. And you can use this to make really pixel-dense heat maps. And so for the dog on the left here, the visualization below, the bright pixels are where, if you change those pixels, the prediction of dog changes most strongly. And sailboats usually are on water if you're doing it right, and so it highlights the sail, but also the water. Whereas the visualization of this jackfruit, I think it is, or something on the right is a little bit more diffuse because it's occluded and it's kind of harder to see. Now the problem with saliency, though, is that if you asked on the left for what are the salient parts of the image to predict a cat, you would end up with a very, very similar visualization. And if you start changing the pixels, you can end up with a completely different heat map. So this is maybe not ideal. And we want to know why the network thinks this is a dog and not just all the possible animals. So another way to do this is class activation mapping, where now you're going to change the structure of your network to include this last linear layer. So in deep learning, we generally love all the nonlinearities we can possibly cram in there. But to interpret, we need something linear, because I think this might be how we operate, as we linearize everything around us. So class activation mapping takes the last convolutional layer and introduces this global average pooling operator, where you take the activation across the whole spatial configuration of the receptive fields that made each filter fire, and you average that all, and then you learn a weight in this next linear layer, and then you can produce heat maps for each of these last layer filters, and if you add them all together, this localizes the Australian Terrier quite well in this image. And actually, to come up with the final probability, you just sum this heat map. The problem with this is that it generally decreases the accuracy of the network, which we don't want. But it does give you a pretty good localization. So we're going to put a bunch of this together, and GradCam picks up on this class activation mapping. And so we've got a sort of vanilla convolutional neural network here. It's predicted cat, and we've got this last convolutional layer, but now we've got some fully connected units here that are pretty common in these kinds of architectures, and we can't do the same kind of global average pooling here, because then these nonlinearities don't work. So instead, we're going to take the derivative of the softmax with respect to all these filters, and then we're going to global average pool that. So it's sort of a combination of a saliency, but not with respect to the input, with respect to this last convolutional layer, and then we're going to multiply those by the feature maps themselves, which we don't have to global average pool directly now. Add them all up, put them through a rectified linear unit, and that's GradCam. And so here, this is where the network thinks the cat is. And this is a hard example because there's also a dog in here. So we ideally want to be able to come up with a prediction that separates the dog and the cat as different things. But this is still a little blurry, and so if you guide it with guided backprop, which produces pixel-dense but not class-specific heat maps, and you multiply these together, you end up with a pretty good pixel-dense, class-specific localization for the cat. And they show that this, for this one example, works quite well. The dog and the cat produce different heat maps, as we expect they should. The dog's up here, the cat's down there, and the occlusion map sort of lines up pretty well with where they predict. Now, I've tried to use this in some never-ending quality control of MRI images, and for this example, I think I might be able to convince myself that maybe the network is learning that there's signal-to-noise ratio that you can pick up in the empty space outside the brain, and if it's pretty clear, that the image is more likely to pass. And the fail image, there's lots of motion artifacts in this, so it picks up on that in some brain areas, but also in the empty space. But really, yeah, I've heard anecdotally that either it works really well and it generates what you expect it to generate, or it doesn't. And it's hard to say when it's right and when it's wrong. And this is tough. So these methods all fit in with the kind of sensitivity with respect to the inputs. But the problem with GradCam, the big problem that Alexander Binder raised, is that they don't model negative evidence at all. So I think most neural networks and most human networks would pretty easily identify that there's lots of positive evidence for horses in this silhouette. But if you change the image just a little bit and add a little bit of negative evidence, now it's pretty clear they're unicorns and not horses. And we want our interpretability method to maybe be able to model this properly. So this was one of the major motivations he gave to layer-wise relevance propagation, where you take a trained neural network and you basically run it backwards, but differently than backprop, by propagating the relevance of the prediction from layer to layer, and it uses these principles of conservation, so relevance can't just appear out of thin air at a layer. It has to be constant all the way to the input. Has to be positive, even though it can model negative evidence. Has to be continuous, so if you change the input slightly, the output shouldn't change that much. And if you remove relevant features from the input, it should reduce the prediction accuracy, which, I mean, these principles all sound pretty good to me, except maybe the continuity one, because, you know, those unicorn horns are pretty small perturbations. And also, layer-wise relevance propagation, there are quite a few hyperparameters in how to propagate through the different kinds of nonlinearities, and what to do with the max pooling and the global average pooling and that kind of thing. And yeah, so there is a ratio of positive to evidence and to negative evidence that you need to use by this alpha and beta parameter. And there's starting to be a lot of these interpretability methods, and I think it could be pretty easy to just kind of try all the combinations until you get a heat map that agrees with your theory, and then you're maybe overfitting your explanation, which we try really hard to not do in the training phase, but I think this is a real risk that not a lot of people are thinking about. There's another method called LIME, the Local Interpretable Model Agnostic Explanations, which has a great GitHub package. And basically, the approach here is to approximate some nonlinear, complicated decision function just locally, just within the space of a single training example, and come up with a simpler model that's going to be a good approximation. We're going to optimize so that we find the best interpretable model G that explains the real model F with some proximity measure that defines what's local and a penalty on complexity. It's all very mathy, but graphically it sort of makes sense if your space and your decision boundary is this blue and red region, and you know, it's hard to come up with a function for this, pretty nonlinear. But for this red cross, this straight line, the dotted straight line, is a pretty good linear approximation to what's important local to that example, whereas a red cross over here that explanation might not hold at all. So basically, you have to generate data to fit this this interpretable model using your proximity measure. And in practice, if you go to their GitHub repository, where this is like 2 lines of code that you can add to any machine learning classifier or regressor, this is what they do in practice. So they just say linear models are going to be interpretable models. We're just going to use the negative exponential of Euclidean distance as a similarity measure, and complexity penalty, we're just going to choose K features. So this nice mathiness sort of disappears in the GitHub implementation, where it's just sort of, they implement feature selection for images, which is not clear, and they take the squared distance to optimize a linear model to fit what the actual model is around the space. So I used this in this defacing detector, which apparently now is not sufficient to detect if an image has been sufficiently anonymized, because now we can reface images with GANs. But if you try out Lime on this, what it actually does under the hood is it groups pixels together into superpixels, where it kind of uses a proximity and intensity to kind of come up with blobs. So I just ran it with defaults here, and so this explanation on the right is the areas that Lime thinks contributed to the correct decision that this MRI image still has a face and is not ready to be shared in the BIDS validator. So it sees part of the face, but it's also picking up this part of the image, and you know, maybe this superpixel approach works for images of cats and dogs and toasters, but is not valid for MRI images or diffusion maps or what you're going to use this for. So this is why it's dangerous, because the GitHub repository is super easy to use. This is literally like 4 lines of code, but you know, you should maybe be careful before you put this in a paper as, you know, explaining causal inference, because there's a lot of assumptions built into all of these methods. So now we've kind of gone over a pretty good sampling of many of the methods that are out there. If I had to recommend one or two, I would pick Lime and GradCam, just because they're super easy to apply to your machine learning. And GradCam really is deep learning specific, but Lime you can use on anything that's machine learning. But you should read the papers if you are going to use them and understand the limitations to the methods. I don't think this is a complete kind of chart of how we can look inside the black box. I think you can design networks interpretably, and so like there's this work showing that spatial transformer networks can be used as kind of like a module of your network and spit out 6 parameters that you apply as a affine transformation to your input, and it just learns registration. So that's like pretty interpretable. There's also ways you can look at the properties of your layers. You can tie weights and build in rotational invariance at different levels, which may or may not be a good things to do, because you know, you might want to do this to make sure that you don't have to do registration, but it's pretty unlikely that the ventricles are going to appear flipped within a brain, or you know, you're going to end up with the corpus callosum halfway across one of the sides of the brain. So you want to make sure that you understand the properties of convnets and GANs and these interpretability methods if you're going to use them. So there's also all the attention stuff that Grace Lindsay presented earlier, which I think is a strong avenue of research, and there's lots of work being done in different directions on this. But I am out of time. So there's also significant confusion as to what interpretability even means. I kind of intuitively think of it
I want to get at causal inference, but you can also talk about it in terms of transparency, simulatability, and there's a great paper by Zach Lipton called The Mythos of Model Interpretability, where he kind of reviews a bunch of the different things we mean by interpretation. So these are kind of the easiest to get up and running with GitHub repositories that are fairly easy to implement on your own. Even if you're not training models yourself, even if you're a total beginner, it's fun to play with, and you do get an intuition just by playing around with other people's code. And I encourage you to go for it. So I don't know if I've given you all of the tools to look inside the black box. And I really want to drive home the message that it is a dangerous thing to try to apply these methods. But I think it is something we need to talk about if we're going to use deep learning to try to do science, because if we can't come up with explanations for why these amazing function approximators are doing the things that they're doing, then maybe we should just leave them alone to the computer science crowd and go back to statistics and try to be rigorous and not just trust in the machine. So thanks a lot. That's all I got, and I'm happy to take your questions. Nobody's got any questions. All right, I have a Colab notebook with a lot of these implemented for the defacing detector implementation. It no longer works because it depends on an old version of SciPy, which is no longer able to be installed in Colab, which I discovered recently. But it's linked to in my presentation, and if you clone it on your laptop, it will work, although you need a GPU for it to be fast. So yeah, we've got a little bit of time for questions for the other presenters. How much time do we have? Oh no, actually we don't. So I'm just going to close the course, I guess, by thanking all the speakers for doing the enormous amount of work it takes to produce a hands-on training, which is really not the same at all as just delivering a pre-prepared lecture. So can we give a hand to the presenters that did this work for us? Thank you also so much to Ariel Rokem, who co-organized the course and did a ton of work making sure that all of the collabs worked and recruiting participants, some of whom I met for the first time today. And I hope that you all continue your journey in deep learning through neuroimaging, and hopefully we can bridge the gap between the deep learning and neuroscience and figure it out what makes us tick, and maybe help some patients along the way. So have a great conference.